Respected Director, RIPS, Dr. J. Surya Rao Sir, Chairman, RIPS, Srimati J. Jayalakshmi Madam, Principal, RIPS, Dr. B.V.V. Ravikumar Sir, Vice Principal, Dr. C.H. Niranjan Patra Sir, today's chief guest, Professor Bimal Krishna Banik Sir, guest of honor of today's program, Professor Dr. G. Devala Rao Sir, eminent speakers, delegates from all over the globe, my dear colleague, and all the great minds present over here. A lovely and warm good morning to a new beginning and a new blessing from the Almighty. I, Rajaram Das, Assistant Professor on the behalf of RIPS, would like to present a gracious welcome to you all to this inaugural ceremony of Series 2 online faculty development program on emerging trends in drug design and development, sponsored by AICTE New Delhi and organized by RIPS. Before starting the inaugural session, let me focus a few highlighting points about this FDP. The major focus of conducting this FDP is to train the faculty members in the field of pharmaceutical education and research. The main objective of this FDP is to update all of us about the new technologies applied during drug discovery and development process in the pharmaceutical research. Drug design is the key step to generate new drug molecules in the drug development. This FDP will help all the faculties to improve their innovative ideas during their research works. It is going to develop technical skills of young faculties while designing new laboratory experiments, project works for the students. This faculty development program is going to give an insight into the field by discussion of necessary theoretical background as well as live interaction with different scientists, academia experts from different industries and research laboratories. To mark this event a traditional beginning, let us begin a virtual presentation of lamp lightning followed by Ganesh Vandana.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
वक्रतुण्डमहाकाय सूर्यकोटिसमप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा
Now it's time. I invite our honorable patrons, Srimati J. Jayalakshmi Madam, Chairperson RIPS, and Director Dr. J. Surya Rao Sir on board for their welcome address.
Namaste to everyone. Most respected chief guest of this occasion, Dr. Bimal Banik from Saudi Arabia, Professor Deval Rao, Principal of KVSR Siddharth College of Pharmaceutical Sciences, Vijayawada, respected Principal Sir, Dr. B.V.V. Ravikumar, the coordinator of today's FDP program, the Vice Principal, Professor Niranjan Patra, other staff members, participants, delegates, and all our well-wishers. Once again, good morning to all of you. For all your kind information, we have successfully completed the Series 1 of the Faculty Development Program being sponsored by AICTE with very successful because we had a set of very eminent persons who have delivered their best. And for your kind information, Roland Institute of Pharmaceutical Sciences was started in the year 1986 with the sole aim and objective is to impart qualitative education in pharma field. And today, in this entire journey of 35 years, we stood on our commitment without any failures. So it is all possible only because I can say that we have got a very good, a very committed, dedicated staff members, including our principal, sir. I thank our principal and staff members for their best possible commitment and dedication all through these years. And I can say that our commitment and dedication is a big strength of RIPS today. I congratulate and thank our principal, Professor B.V. Ravikumar, who is the coordinator for Series 2 of the Faculty Development Program, and also Dr. Biswamohan Sahu, the co-coordinator to this Faculty Development Program. Thanks to AICTE for funding this project, and hope that this Faculty Development Program will be immensely benefited. I feel privileged and honored to see very eminent personalities like Professor Bimal Banik sir from Saudi Arabia and also Professor Deval Rao from Vijayawada. Sir, you have accepted our invitation and spared your valuable and quality time to spend with all of us on this webinar, online webinar. So it is indeed a great privilege and pleasure on my part to thank you very specially from the bottom of my heart. With this, I welcome our chief guest, Dr. Bimal Banik sir, and also Dr. and Professor Deval Rao sir for this accepting as the guest for the Series 2 of this Faculty Development Program today. A set of wonderful resource persons are there to deliver their best in this second series. Also, it will be of great use to each and everyone. I am very much confident that all the participants will definitely abreast with this latest knowledge in this pharma field too, in this pharma field very especially. Wishing this pharma, the Series 2 of this Faculty Development Program a very grand success. Once again, I thank you all. Thank you for giving me this opportunity to welcome all the dignitaries for today's inaugural session. Thank you all.
Thank you so much, madam, for your constant support and encouraging words. Now I take this opportunity to invite our respected principal, Professor Dr. B.V. Ravikumar sir, coordinator of this FDP, to address the gathering.
Thank you, Mr. Rajaram. Respected chief guest for today's function, Dr. Bimal Krishnan Panik sir, Professor and Dean, Prince Mohammed bin Fahd University, Kingdom of Saudi Arabia. Respected guest of honor, Professor G. Deval Rao sir, Principal, KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada, Andhra Pradesh. Respected Director, Dr. J. Surya Rao sir, Chairperson, Madam J. Jayalakshmi, Co-coordinator for this program, Dr. Biswamohan Sahu, Vice Principal, Professor Niranjan Patro, all the faculty members of RIPS, and all the resource persons of this Series 2 Faculty Development Program sponsored by AICTE.
Everyone can see
And my dear delegates, at the outset, I welcome all of you to this Series 2 faculty development program sponsored by All India Council for Technical Education. We have successfully completed the Series-1 of Series-1 FDP in the last month, and we have received a overwhelming response from the participants, and the feedback was also very good. I thank all the resource persons of FDP 1 series for their uh, time and sharing knowledge with all the participants. Now, I would like to congratulate and thank all the delegates of this Series 2 FDP for showing interest in this program. And I also like to thank principals of all the delegates for allowing them to participate in this faculty development program. As our chairperson madam told, this FDP is lined up with lectures by eminent persons from industry and academia, not only from India but also from abroad. I thank all the resource persons for sparing their valuable time to enrich the knowledge and skills of all the participants. I appreciate Dr. Bisumon Sahu, co-coordinator for this program, for his efforts to make this FDP a successful one. I would also like to thank Professor Niranjan Patro, Raja Ramdas, Jasma Jana, and entire faculty members of RIPS for their support. So we are doing all these activities with the support of our management. I would like to thank management of Roland Institute of Pharmaceutical Sciences, Chairperson Madam Jayalakshmi, and Director Dr. J. Surya Rao sir, and all the governing body members for their constant support and encouragement. I also like to thank AICTE for funding this program. Last but not the least, I would like to thank our today's chief guest, Dr. Bimal Krishnan Manik sir and Professor G. Deval Rao sir for sparing their valuable time to grace this occasion and to motivate all the participants who are participating in this FDP. With this, I thank the organizers for giving this opportunity. Thank you very much.
Thank you so much, sir, for your all-time support and constant monitoring. I would now like to invite our respected Vice Principal, Professor Dr. Niranjan Patru, sir, for sharing his views about this FDP program.
Am I audible?
Yes, sir, you are audible.
Good morning, all the respected dignitaries present on this online platform. I would like to congratulate our principal, Dr. BBV Ravikumar sir and Dr. Visomon Sahu for starting the second series with again a very good number of resource persons. I am sure that this program is also going to be successful as the series 1. The backbone of any educational institute is teachers, and if the teachers are updated with such kind of faculty development programs, that will be reflected to the students, and students will get good knowledge, and in turn that will give a good result for the society. So the basic objective of this program is to enrich the knowledge of participants, that is mainly the teachers and the research scholars. So my best wishes for the program, and thank you very much for giving me this opportunity. Thank you all.
Thank you so much, sir, for your motivational and inspiring words. Roland Institute of Pharmaceutical Sciences, since the year 1986, has been continuously adding many success stories to its name, from being one of the best pharmacy colleges in Odisha and many more tags to its name. Now it's time to hear from our guest of honor, Professor Dr. G. Devala Rao, sir. Before inviting sir for his talk, I will take this opportunity to introduce Professor Devala Rao sir to all of us. Dr. Devala Rao Garigapati sir, Professor and Principal, KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada, Andhra Pradesh. Dr. Devala Rao sir is a well-known facet whenever the word pharmaceutical analysis comes to our mind. Sir is having a teaching and research experience of more than 32 years till date. Dr. Rao sir is the approved PG guide by Acharya Nagarjuna University, Nagarjuna Nagar, and Krishna University, Machilipatnam. Sir is approved research guide by JNTU Hyderabad, JNTU Kakinada, JNTU Anantapur, Acharya Nagarjuna University, Sri Krishna Devara University, Sri Chandra Sekharandra Saraswati Deemed University, and Christ University. Sir is involved in the development and validation of analytical methods for different categories of therapeutic agents. Based on his research work carried out, Sir has published so far 375 research papers in various national and international journals of repute. Sir has guided more than 30 candidates for PhD and 15 candidates for MPhil degree, leading to their completion. Presently, around 15 candidates are pursuing their research work for their PhD degree. Sir is also appointed as the Inspector for Pharmacy Council of India, New Delhi. Dr. Rao is the ex-chairman, Board of Studies in Pharmacy from Acharya Nagarjuna University. Sir is the chairman, Board of Studies in Pharmacy, Krishna University. Sir is the academic senate member for Krishna University, nominated by Government of Andhra Pradesh. Sir is the member CFO AP Pollution Control Board. Sir is the member of the Task Force AP Pollution Control Board. Sir is the member of SEAC, Ministry of Environment, Forest and Climate Changes, nominated by Government of India. With this brief introduction, it's my privilege to invite Dr. Devala Rao sir Garigapati on board.
Thank you, Dr. Rajaram. Thank you very much for your kind introduction and kind words. Good morning, one and all. First of all, I would like to thank the management and faculty members of Roland Institute of Pharmaceutical Sciences for inviting me as a guest of honor for this second series of AICTE sponsored emerging trends in drug design and development. So on this happy occasion, I would like to thank Srimati J. Jayalakshmi Garu, Chairperson of Roland Institute of Pharmaceutical Sciences, Dr. J. Surya Rao Garu, Director, Roland Institute of Pharmaceutical Sciences, Dr. BVV Ravikumar, the Honorable Principal of Roland Institute of Pharmaceutical Sciences, Dr. Niranjan Patro, Vice Principal, and my good friend and co-coordinator of this AICTE sponsored workshop, Dr. Desamohan Sahu, and the faculty members of Roland Institute of Pharmaceutical Sciences, Berampur, and all the delegates who are participating. And finally, the most important is chief guest for today's second series is Professor Dr. Vimal Krishna Baniko, Professor and Dean Prince Mohammed bin Fahd University, Kingdom of Saudi Arabia. Very good morning to all of you. So first of all, I am very happy to associate with all of you because this kind of what is known as the faculty development programs or refresher programs or whatever name you give, usually these kind of programs are not available when we are students actually at Andhra University due to various reasons that we don't want to dig now. But now the present generation, they are very lucky. They are having what is called this kind of what is known as the events organized by reputed institutes like Roland Institute of Pharmaceutical Sciences with the support of funding agencies like AICTE or some other agency. So in this regard, I would like to congratulate all the delegates because from the introduction words of Dr. Ravi Kumar, what I understood is the first series is successfully completed, and now they are starting what is called the second series day after tomorrow, on 17th, my talk is there from 12 to 1. I was invited by Dr. Mohan to deliver a talk, and I happily accepted. My talk will be there on 17th, the day after tomorrow. So usually, these kind of what is called the programs, they are very important for what is called the young minds, especially the teachers who joined recently. Because generally, I think all of you know, if we take what is known as professional colleges like pharmacy or engineering, or if we take what is called the other colleges, by virtue of qualification only, we are not teachers by profession. By virtue of qualification, maybe M.Pharm or PhD or some other degree, we are all entering into this what is known as the noble profession, and it is a very important profession. Therefore, what is important is each and everyone, you should know how to deal with what is called the students. That is very important. Therefore, as a what is known as the senior faculty member, what I can tell is usually this kind of what is known as the training program is highly essential for any youngster, any young teacher, because only when you are having what is called training programs, then only we will get what is called some ideas. We will come to know about what is called emerging trends or new techniques or what is called the new teaching methodologies also. After that, due to this what is known as the pandemic COVID-19 for the last 1 year, I think all of you know, we are all shifted to this what is known as the platform that is called virtual platform. Thanks to this what is known as the pandemic situation, because of this, we all moved to this what is called the virtual situation forcibly, definitely, no doubt. And it is a wonderful platform because myself, I am in Vijayawada, Sari in Saudi Arabia, you are all in Berhampur, and delegates are from different places throughout the India. But even then, we are all meeting and we are all sharing our what is known as the views. So on this application, what I feel is definitely in the series 2 also, we are having what is called a very good mix of what is called talented teachers, experienced teachers, or what is known as the highly qualified teachers. So my sincere appeal is please utilize the what is known as the lectures or what is known as the advices. They are going to be given by what is known as the senior faculty members. So as a part of this, what is known as the AICTE sponsored, it is known as the program. AICTE sponsored, it is known as online faculty development program. That is emerging trends in drug design and development. Now, if we take this drug design and development, I think all of you know, even though it looks like a very simple word, it is not so simple. It involves what is called two things, that is design and development of drugs. So there is a lot of what is known as the process involved in the drug design and development. We know because for the last 1 year, we are all in what is known as pandemic situation. Luckily, thanks to what is known as all the pharmacists or scientists, doctors who are in India, they have developed successfully the vaccine. Now, vaccine came. But how the vaccine came means it is not so simple with the what is known as the day and night effort of all the pharmacy people, all the scientists, all the doctors, all the microbiologists, all other what is known as the people involved in this what is called the noble activity. I can say they are all warriors. We are able to get what is called it is known as the vaccine, and you know it is in the third stage. Actually, third stage is successfully completed. But even then, people are taking in a what is known as the batch-wise manner, and because of the God's grace, the vaccine is working wonderfully. Definitely, very soon, all people in the country will be vaccinated with what is known as a corona virus vaccine, so that definitely we will be free from what is known as the virus. For all this, what is important is always you should learn what is known as new methodologies, new techniques, new trends. They are called new trends, emerging trends. So how we know this kind of trends means by organizing this kind of what is known as the seminars or faculty development programs or staff development programs. Usually it is possible to provide what is called the good knowledge. So on this happy occasion, once again, I congratulate the management, especially Srimati Jayalakshmi Madam Garu, chairperson, Dr. J. Surya Rao Garu, director, and the principal, vice principal, faculty members, and other members for their what is known as the noble efforts in arranging this kind of what is called a series of programs. So series 1, series 2, series 3 means why they are arranging what is called in the 3 different series means the people who missed in first series, they may join in second, or those who missed in first and second, definitely they will get a wonderful opportunity in the third one to participate and to get what is called knowledge from this what is known as the workshop. So with these kind words, I am very happy with what is called participation in this Roland Institute of Pharmaceutical Sciences, because I know it is a very old college. When I was a student at Andhra University, 82 to 87, BPharm, 87 to 89 at M.Pharm, Andhra University, some of my seniors, they used to work in Berhampur actually. From that time onwards, I know what is Roland Institute. So several of my friends, my roommates, my classmates, they worked at Roland. I know it is a very noble institute and a very highly rated institute in Odisha, and they are doing this program, means very happy. And similarly, my friend Dr. Visamohan Sahu, he worked in Vijayawada for a few years actually. We moved very closely. I used to go to that college as a guest or as a speaker for several occasions. So in that way, he is maintaining very good relation. Usually in a professional field, what is important is whenever you met some personality in your life, my request is don't forget, just try to maintain the relation. That is the only way how you can maintain what is called good relation wherever you may be. Maybe in India or abroad or some other place or some other state, but definitely we'll get what is called good interaction if you maintain what is known as the this kind of what is called the relation. And once again, thanks, and thanks for giving me this opportunity. Thank you, Surya sir, also. Thank you, sir.
Thank you, sir.
Thank you. Thank you so much, Professor Devla Rao sir, for accepting our invitation and gracing this faculty development program with your presence. We have with us our esteemed chief guest of today's event, Professor Dr. Bimal Krishna Panik sir with us. Before inviting sir for his talk, I will take the opportunity to introduce sir to all of us. Although Professor Panik don't require a formal introduction from our pharmacy fraternity, Professor Bimal Krishna Banik sir has conducted doctoral research at the Indian Association for the Cultivation of Science. Sir has conducted postdoctoral research at Case Western Reserve University, USA, and Stevens Institute of Technology, USA. Professor Banik was a tenured full professor and first President's Endowed Professor at the University of Texas and the Vice President of Research and Educational Development of Community Health Systems of South Texas, United States of America. At present, Professor Bani is the professor of the Deanship of Research Development and College of Natural Sciences and Human Studies at the Prince Mohammed bin Fahd University in the Kingdom of Saudi Arabia. With this formal introduction, it's my great privilege to invite the chief guest of today's event, Professor Panik sir, on board.
Namaskar. Bisho,
You can, sir, you can share your screen.
Dr. Bisho,
Sir.
Dr. Bisho,
Sir, it is audible, sir. Tell me, sir. Sir, you just
can you
go
hear
to share
me?
screen. Yes, sir. Sir, your network is somewhat slow, sir, I think.
Can you hear me?
Yes, sir. Audible, sir. Sir, can I share your screen otherwise?
Can you hear me?
Yes, sir. Audible, sir.
Bisho,
Sir, audible, sir. Audible. Sir, it is audible, sir. Sir, contact one, sir.
Bisho,
Sir, it is audible.
Bisho. Hello?
Sir, sir, I am sharing your screen otherwise, sir. Right, sir. You can start now, sir. You can start, sir. Sir, I have shared your screen, sir. You start now.
Hello, can you hear me?
Yes, sir. It is audible, sir. I have already shared your screen.
Okay, but there is a problem. I am getting disconnection again and again. All right.
Sir, you can.
Should I start now, Bishop?
Yes, sir. Yes, sir. Continue, sir.
Okay.
Thank you, sir.
First of all, good morning to everybody. I am very grateful to the organizer for giving me an opportunity to discuss something related to drug design and development. Ladies and gentlemen, this is the extremely hot topic. I don't know I know anything about drug design and development. This is the most complicated subject in the world. Even a simple molecule, when we take as a medicine, many expert scientists do not know what is happening in the body, in human body and animal body. Perhaps the most complicated task in the world is to design drug and to develop it. In our case, some of them are extremely complicated, and obviously many of you are expert, many of you are learning. Your time is very important. I can create a lot of controversy if I say something and if I do not say something. If I say something, you may feel I am telling something which is not good enough. And if I do not say something, you may feel I am not telling you the truth. Vishwa, can you forward me next one? We design molecule, drug, based on the type of disease, nature of disease, and the propagation stage. Bishop, forward next one. Next. Drug discovery process started many years ago from herbal drugs, vitamins, new technology, breakthrough in chemistry, biology, pharmacy, clinical science, health, drug discovery program. Vishnu, next. Out of 10,000 compound, only one particular compound can become a medicine. So you understand the complexity in the subject. And this is American statistics. Out of 10,000 compound, one became medicine. So you can imagine the situation of India. India has advanced a lot in many disciplines. However, being an Indian, originally from India, I must tell you that drug discovery project is not good enough in Indian science. Several years ago, I delivered a lecture in Indian Science Congress, 100-year anniversary. There I pointed out this fact. Since then also, I see mostly we do contract research. Means somebody, the drug company gives offer to the Indian company and they prepare the molecules drug for them. They don't develop drug of their own. This is the limitation we have in our country because of the fact out of 10,000 molecule, one can become medicine. Vishnu, next. Vishnu, next. Bisho? So probably system is not working properly. Bisho? Hello, Bisho? Skinning, manipulation, designing, metabolite, and serendipity. Serendipity also helps in some occasion. These are all equally important. Nobody can say this part is more important than the other. All stages are very important. Next. Next. To do drug discovery, drug development work, we need a lot of items. That's why this subject is complex. Chemistry, biology, medicinal chemistry, clinical biology, pharmaceuticals, and clinical trial. And these are all important, and that's why it is very time-consuming process. Bisho next. Bisho next. There are many phases of drug discovery process. Phase 0, 1, 2, 3, and then clinical approval. For this, you know that expertise of different level of scientists is important. It is not only one single individual. It is a group. It is a team that can develop medicine and drug. Okay, Bisho next. That's why it is phase 1, and different types of trials are important to identify molecule as drug. Bisho next. Phase 2 clinical trial particularly is important to identify the safety, efficacy, toxicity, and drug interaction. These are important because a molecule may be very good to cure or to stop some particular disease, but that can create other side effects. That's why some molecules are good enough in terms of activity against a particular disease, but the same molecule may create other disorder. Therefore, that molecules or drug cannot be used as a drug candidate or even as a drug. That's why it is a problem. We have many problems we see because of the designing. We show next. Clinical phase 3 clinical trial are very important also. Some drugs can pass phase 0, 1, phase 1 and phase 2. They may fail in phase 3. Many molecules may not enter from the program. Therefore, drug discovery is complicated and troublesome process. Okay, be shown next, please. Type 4 is mostly marketing and monitoring. And phase 4 is relatively simple than phase 1, phase 2, phase 3, because phase 4 is mostly business related. At that time, we know most of the facts about medicine or drug development or design. Next. Bisho next. Bisho next. Gene therapy is a modern term. Still, it is discovered years ago. And gene therapy is very important term, and that is used in drug development and science. Bisho, go please quickly because I have limited time. Because you gave limited time. Bisho, please go quickly. Next, please. Many scientists are involved in students. Bisho, next. System does not work properly. Yes, Bisho, please go to the next. Next.
It is over, sir.
Okay.
Completed.
In my this talk, I wanted to say something related to collaboration. The young generation scientists, you must collaborate with others. For example, you are a pharmacist. If you feel you will be working only with pharmacy people, you will not be able to develop any drug. I challenge. Similarly, you are a biologist, you live in fool's paradise. So, drug discovery efforts is a combination of work among chemists, biologists, physicists, computer scientists, and so on. It is a complicated process. It is a frustrating process, but still we have to do. If we can select, if we can make from 1,005 molecules as drug, that will be wonderful. Our challenge is to identify new methods, new entity, new molecules, new synthesis, new pharmacokinetics, measurement, detection, even some molecules which are disregarded as the medicine at this time, whether we can do manipulation of that molecule and make the molecule more effective for different types of disease. One molecule is designed for a particular type of disease may not be very good for that disease. It may be effective for other disease. So one should not rely on one particular disease only. So that molecule, when people find some activity against a disease, that molecule must be tested against other medical disorder. It has been found molecule designed for a particular disease has been used for other type of disease in the future. And with this note, because of time limitation, I conclude my lecture, and I thank the Roland Institute, all the listeners, and Government of India for supporting this kind of program. And this kind of program, I don't know, I have never participated in this kind of program. I wish all success. I believe from this program, young generation can learn a lot of things and can apply in their future without staying in their boundary. They should move out from their comfort zone, and they should collaborate with people regardless of their discipline. That can help drug discovery and drug development work in the future. Thank you very much for your attention.
Thank you, sir. Thank you
you.
for your nice presentation.
Thank you, Professor Banik sir, for enlightening us with your great knowledge. Your words are definitely going to help us in refining our research activity. May I now request our esteemed chairperson and patron of this FDP, Srimati J. Jayalakshmi Madam, to kindly present a e-memento to our guest of honor of today's event, Professor Devalarao Garigapati sir, as a token of our love and affection.
Thank you, Devalarao sir, for sparing
Thank you very much,
time.
sir.
We heard about you. I know you, sir. You are a classmate of MBV Raju, sir. No, sir?
Raju is my classmate, sir.
Yeah. He used to tell about
And
you.
Nagoji is my roommate, sir.
Okay, sir. And
Might be knowing Nagoji also. And Raju is my classmate, sir.
yes, sir. Both worked here, sir, in RIPS.
I know, sir, actually. Butchengat Batraju is my classmate.
Okay.
So, he's roll
We
number
read
4
your
in my.
books also, sir.
Yes, sir. Okay. Ravi Kumar, sir, you are from Andhra University?
Yes, sir. 98 pass out, sir.
98 pass out. 98 means almost. Okay,
Yeah, 6 years.
we passed out in 89, sir, long back, almost 10 years gap.
Yes, sir.
So you are analysis, sir, chemistry, acidic, sir?
Chemistry, sir.
So chemistry means Professor D. Venkatrao, sir, BSS, sir, Shastri, sir.
D. Venkatrao, sir, is my guide, sir.
Sir is my our favorite teacher, sir, in chemistry, Andhra University, means D. Venkatrao, Professor D. Venkatrao. Recently, I was spoken with him, sir, 1 month back.
Okay, sir.
Spoken with
Thank
the
you, sir.
madam.
1 second, sir.
Thank you.
Ma'am, please hand over the memento to the sir.
Sir, please accept the memento and certificate from
Thank you.
Roland Institute of Pharmaceutical Sciences, Berhampur. And we are
Thank you.
extremely
Thank you very much, madam.
We are extremely thankful for you for your presence on online webinar. Thank you very much, sir.
Thank you very much, madam. Thank you. I'm really blessed. Thank you.
Thank you, sir.
Okay, thank you, Mr. Mohan. Okay.
Thank you, sir. Thank you, sir.
Okay, thank you. Thank you.
May I now request our esteemed chairperson and patron of this FTP, Srimati J. Jayalakshmi Madam, to kindly present a e-memento to our chief guest of today's event, Professor Vimal Krishna Banik sir, as a token of our love and affection.
Sir, as a token of our love and affection and memory, from the team RIPS, we present you this memento and certificate as an honor to be our chief guest for today's inaugural function for Series 2 of this FDP being sponsored by AICTE. Thank you very much, sir, with your presence.
Thank you, sir. Please.
Thank you. Namaste. Thank you very much for your kindness. I sincerely accept it from you, ma'am. Thank you. On behalf of my university and on behalf of my family, I sincerely accept it. Thank you. Namaste.
Thank you.
Namaste.
Thank you, sir. Sir, we will give short 5 minutes breaks, then we'll start your scientific session.
Bisho is
5 minutes. Sir, you can start your scientific session within 5 minutes. Sir, I will share your screen, sir.
Finally, I would like to take this opportunity on the behalf of entire RIPS management to place on record of our hearty thanks to the chief guest, Professor Dr. Bimal Krishna Banik sir, and the guest of honor of today's program, Professor Dr. Devalaro Garigopati sir. We are really enlightened with your knowledge and presence, sir. We are thankful to our Honorable Director Sir and Chairperson Madam for their constant support and motivation in all dimensions. I'd like to thank our Principal Sir and Coordinator of this FTP, Professor B.V. Ravikumar Sir, for his enthusiastic support. A special thanks to the co-coordinator of this event, Dr. Bisumon Sahu Sir, for his timely arrangement and his precise efforts for the smooth conduction of this event. My special gratitude to all the members of organizing committee, teaching and non-teaching staff members, eminent speakers from around the globe for sparing their valuable time, and all the delegates for their unflinching support and coordination. With these warm words and kind messages from our chief guest and guest of honor, we now officially conclude the inaugural ceremony of the Series 2 online FDP program sponsored by AICTE New Delhi. We'll be resuming back with the first session of our eminent speaker, that is Professor Bimal Krishna Banik sir, very shortly within a span of 5 minutes.
Sir, I'll share your screen, sir, from my side.
Yeah, there's problem from your side or my side?
No, sir. Just 5 minutes break. We'll start, sir.
Oh, okay. I do not know that.
Okay.
Okay.
Just I'll share, sir. Just within 2-3 minutes.
Because system is
Sir, I'll share the screen. No
disconnecting
problem.
again and again.
Sir. We'll start, sir.
So another 3 minutes.
Sir. Just 2 minutes. We'll start, sir.
Okay, but what I'm saying, why the system is disconnecting? Okay.
The network problem is there, maybe.
Okay, 2 minutes. Okay.
Sir, we can start now.
Okay, start. Good afternoon and welcome back, dear delegates and participants, to the Series 2 of online FDP program sponsored by AICTE. We have with us our international resource person, Professor Bimal Krishna Banik sir. Before starting his eminent talk, let me first of all introduce sir to all of us. Professor Banik has been involved in chemical and medicinal research on diverse cancers, fertility hormones, infections, catalytic reactions, green and sustainable approaches, and microwave-induced exploratory processes. As the principal investigator, he has been awarded over 7.5 million USD grants from US NIH and US NCI. Importantly, Sir has published more than 490 peer-reviewed papers, patents, reviews, perspectives, books, and book chapters. Moreover, he has approximately 500 presentation abstracts. A number of his presentations to the public and scientific conferences are considered as distinguished, featured, keynote, endowment, special, and educational research. And the number of citations of a research paper is over 6,750 with 45 H index. His research contribution and recognition has resulted in media exposure approximately 200 times. Sir has presided over 20 research sessions at the American Chemical Society national meetings and over 2 dozen conferences at the state and international level, including one at the Nobel Prize celebration meeting. Sir has Sir is a reviewer of 93, editorial board member of 28, editor-in-chief of 12, founder of 5, associate editor of 4, and guest editor of 10 international journals. Sir has recruited approximately 250 PhD-level associate editors, regional editors, and editorial board members from different countries. Sir is an examiner and panel member of US NSF, US NCI, ACS, and international grant applications. Over the years, Sir has served as the chair of member of more than 100 scientific committees. Professor Banik has taught several thousand students in the class in USA universities and Saudi Arabia University for the past many years with exceptional skills. Sir has mentored over 200 students, 27 postdoctoral fellows and research scientists, and currently advising 22 university faculty members in their research work. Dr. Banik has acted as the advisor of 2 societies that had more than 1,000 number of students. With this brief introduction, let us hear from Professor Banik sir regarding his talk upon development of polyaromatic compounds as anti-cancer agents. May I now request Professor Banik sir on board to deliver his eminent talk.
Sir, you can start now. Sir, your presentation is shared already. Sir, am I audible, sir? You can start now. Sir, please start now. I do not see PowerPoint. These displays are already on screen. Now, okay, sir, it is visible. So maybe network problem is there, sir. Network was problem. So now it is visible, sir. Screen is visible to you, sir. First I change the slide, you can see, sir.
Can you hear me?
Yes, yes, sir. Audible, sir. You are audible. You can start, sir. Please start, sir.
Go back. Bisho, go back, please. Go to the first one.
Sir, sir. Please start, sir. Now.
Can you hear me?
Yes, sir. Audible. Yes, sir. Audible. Audible, sir. You can start.
Can you hear me?
Yes, sir. Yes, sir. You can start, sir. नेटवर्क चला जाता है Sir, it is visible now, sir. You can start, sir.
But its system is not stable. It disappears again and
Oh,
again. Should I start
okay.
now?
Yes, sir. You can start, sir.
Okay, thank you. I hope
Okay,
it
sir.
works. I'm very sorry because this is
Yes, sir.
giving a lot of trouble to me and Dr. Bisho also. And
No, sir.
probably it is because of the line. I do not know.
Sir, sir.
Let me, Bisho, you have audience?
Yes, sir. Already there, sir.
Okay, because I cannot see anybody.
Yes, sir. You can continue.
Okay, let me start.
Sir.
I thank Roland Institute of Pharmacy Department for giving me a chance to speak something about our research. I am very proud of you. Today I'll talk about polyaromatic compounds as anti-cancer agents. Polyaromatic compounds are mutagenic. They promote cancer. Here I am saying polyaromatic compound as anti-cancer agents. That means I am contradicting the existing literature. I am saying opposite. In contrast to other discoveries, I am telling polyaromatic compound can work as anti-cancer agents. The anti-cancer activity of polyaromatic compound and the cause of their activity definitely is different than polyaromatic compound that cause mutagenic effect or carcinogenic effect. All of us know some polyaromatic hydrocarbon, particularly benzopyrene, is extremely carcinogenic and that can generate different kinds of tumor in human. We show next one. Next. We show, I'll tell you next, like this one, next. See, here we have many compounds, phenanthrene, dihydroanthracene, chrysene, dibenzofluorine. These are polyaromatic hydrocarbon. And here, using this kind of polyaromatic hydrocarbon, we prepared a number of derivatives, a number of new molecules, and these new molecules have demonstrated anti-cancer activity in vitro and also in animal model. Particularly here, the derivative structure AR means polyaromatic nucleus and a two amino component connected to the aromatic system. And this side chain has different kinds of group at the terminal site of the molecule. The terminal site and the side chain has a lot of influence on the biological activity, pharmaceutical activity of these molecules. Next. Next. We have seen some polyaromatic compounds that demonstrated good medicinal activity many years ago. Some of them are shown here. These molecules have extremely potent pharmaceutical activities, and their biological action is documented in literature. Next. Here we see some molecule, and these molecules are studied by USA and very famous scientist Bayer. Here we see polyaromatic compounds are substituted with amino alcohol. And this kind of compounds have demonstrated some sort of anti-cancer activity in vitro. However, these studies ultimately did not proceed because of some activities, some properties which we did not find in literature. What we see that these molecules, scientists studied and they found some activity. Because of these results, we became interested in developing anti-cancer agents using polyaromatic ring system. Next. Here, I hear some polyaromatic compounds derived from naphthalene and anthracene. are shown. These molecules are active against cancer through DNA intercalating properties. Anti-cancer molecules can be active because of different kinds of properties. DNA intercalation is only one of them. One particular molecule can be DNA intercalated. The same molecule can act because of other types of action. So, multiple mechanisms of anti-cancer molecule may exist. I'll come to them slowly. Next. Now, our study. What we have done, we selected chrysin, a tetracyclic aromatic system, to functionalize this aromatic hydrocarbon. We first attempted to nitration because the nitro functionality can be converted to something else, for example, amino. This amino chrysin was prepared by nitration and then by reduction. Next. To this nitro and then amino compound, we added side chain. This piperidine and piperazine containing side chain. We have amide structure in A, we have amine structure in B. We'll see the difference of their bioactivity. Next. Next. This amide group can be reduced by lithium aluminum hydride to the amino group. Now, next one, sir. Here, we prepared similar molecules, but in these molecules, we have kept another type of functional group at the ring system. This functional group may be electron donating, may be electron withdrawing. We also know the electron donation and electron withdrawal of molecule can have tremendous effect on the medicinal activity. So therefore, we prepared methoxy amino nitro bromo acetyl derivative of this diamide and diamine to study their biological activity, anti-cancer activity in great detail. Here I am showing the synthesis of methoxy diamide using chrysin as the core molecule. Next. Here I am showing the synthesis of acetyl derivative of the diamide. This acetyl group has gone to 12th position of the ring. Acetyl group is electron withdrawing. The previous one, methoxy, is electron donating. Now we have fulfilled the game of non-substituted compound. Now one electron donating compound and one electron withdrawing molecule. Next one, Bishop. Here I have bromo. Bromo halogen, sometimes chlorine, bromine, iodine. The substitution of the aromatic ring by halogen may have very good properties. So we substituted the aromatic system by bromine also. Next. See, we have the bromine. We can do the bromination directly with the chrysin system or keeping the side chain in the molecule. Two ways we can perform the molecule. By this process, we can introduce the bromo in the aromatic system. Next. Here, the attention I told you before that anti-cancer drug can act through DNA intercalation. DNA intercalation properties, and this has been documented in scientific literature extensively. In contrast, when you have many hydrophobic molecules, for example, chrysin or polyaromatic compounds are hydrophobic. They repel water. This hydrophobic molecule may not go to the nucleus of the cell because they are hydrophobic. And when a person considers the structure of the cell, many scientists, even today, ignore the surface of the cell. When cancer chemotherapy is involved, scientists always focus to DNA, RNA, protein. Many people ignored the interaction of the cell with the molecules in different pattern. One pattern is the cell membrane. And this hydrophobic molecule, because of their structure, we thought they may demonstrate anti-cancer activity through their surface, through their membrane. To hook this molecule to the membrane of the cell, we adopted a policy, a technique by which we can keep certain polar group at the terminal site of the molecule. This polar group then will bind to the phospholipid, the negative component of the protein present in the cell, and they will hook up there. And that's why by hooking up this molecule to the surface, to the membrane of the cell, one can demonstrate anti-cancer activity. To have polar group at the terminal site, we adopted this scheme. The carboxylic acid was prepared first by doing a simple chemistry, well-known chemistry. Show next. Now you see we have a similar molecule what I presented to you before. Now I have a polar group at the terminal site. OTMS can be converted to OH. This hydroxy derivative, because of the polar nature and because this hydroxy is present at the terminal site of the molecule, this amide we show next. Yes. Now the the OTMS group is deprotected to alcohol, and this alcoholic group, this is the polar group at the terminal site. This molecule can act anti-cancer activity through the surface, through the membrane of the cell. Bisho next. Here we see that we have polar group NH group at the end, terminal site. So we have the access to prepare different types of polar hydroxy amino component at the terminal site of the resulting molecule, because this molecule can be tested to find out whether this molecule can exert anti-cancer activity through cell membrane interaction, which is not widely studied. Other interactions are studied widely. I will come to this now. Bisho next, please. Here are all the examples I have shown you before. These have only one chain. Now I want to demonstrate that scientists can make double chain polyaromatic compounds. There are meaning of double chain because when you consider anti-cancer drugs, you can see bis intercalation. Bis means double intercalation. Double intercalation or bis intercalation can help the molecule even more effectively. Therefore, we prepared bis compound, double side chain containing compound, using a strategy which is dinitration, diamino, and then coupling of the diamino with the side chain. Bisho next. Here, I am showing a fantastic example of bis compound. Here you see two chains are present in the chrysin system, and these two chains have amine and amide. Now we have double chain in chrysin. Now I will show you the medicinal activity of these molecules very soon. Bisho next. Now our job was to change the aromatic core. Earlier, all the example I have shown you with chrysin, phenanthrene, and anthocene system. Now I am showing you dibenzofluorine system. Dibenzofluorine system is different than chrysin system or anthocene system because this system is angular in nature, and because of this angular geometry, the molecule can penetrate to the DNA helix much more effectively, because this angular nature of the molecule is like a finger nature of the molecule. All fingers are not same. If all fingers are same, the way the molecule, the finger will go to the cell is different than the way we have. That's why this molecule was prepared, and then the same side chain was added following the same chemistry technique. We show next. So here we need amino compound. We prepared amino compound here using dibenzofluorine. We show next. Now we have installation. We install the side chain that we using the chemistry and the reaction what I developed before. Now you see that this system and the side chain which is same. Now this system is different. That's why DNA intercalation properties or enzyme inhibition properties or the cell membrane properties will be different. Next. Next. Here we have the same system, but we have one added methyl group. And using this methyl group, we prepared the similar molecules to evaluate them against different kinds of cancer cell. I'll come to now. Wait for a minute. Bisho next. Bisho next. Here, that's the thing you are interested in, because many of you are pharmacists. You do not like to develop chemistry, but without chemistry, you cannot have any molecules. So to make molecules, then only you can do testing. Now I'm showing you the results, biological anti-cancer results of the resulting diamide and diamine using chrysin. Here I am showing the results, vitro results, cell growth inhibition data against different cell line. B16 melanoma, BRO melanoma, HL60 leukemia, L1210 cancer, blood cancer, NCF7 breast cancer, OPCAR3 ovarian cancer, PC3 prostate cancer, HT29 colon cancer. What you see, the cisplatin data and our molecular data, molecule we made, have comparable IC50 data. And interestingly, you see the selectivity. The compound group number 1A practically has no activity, and compound group number 1B has activity. Tremendously high activity has seen in compound 1B, no activity has seen in 1A. Compound 1B has similar properties like well-known medicine cisplatin. Cisplatin is one of the highest selling medicine in cancer chemotherapy work. Bisho, next. Next, Bisho. Bisho, next. Here, using the dibenzofluorine system, we see the activity of the two series. This molecule 1G and 1H they're highly active against all types of 7 cancer cell line. And in fact, they have comparable activity with respect to cisplatin. So starting from polycyclic aromatic compounds, we have demonstrated here molecules that have comparable activity like cisplatin. And I will tell you after a few minutes that this anti-cancer activity is not because of carcinogenicity. Carcinogenic molecule will kill all cancer cells, creating toxicity. Bisho, next. Now, I'm showing some in vivo results. In animal model vivo, against some types of cancer cell. We cannot do all types of cancer cell because this is very, very expensive and also money dependent. In America, no institution gives money to the faculty to conduct research. This is competitive. Faculty have to earn money to support scientists, postdoctoral fellows, students, visiting scientists, technician on all cost. So we cannot do all states. I'm fortunate I could do some. You see, the breast cancer propagates because of stage. When we identify the cancer at the time, we can do the diagnosis. Earlier we identify, better is the chance for cure. We know that. That's why you see here when it's stage 3, survival rate is 93%. Stage 2, stage 3, 72%. After 70, stage 3 is very difficult to survive. Next one. This cancer comes because of some duct present in human breast. Normal duct, when you do electron microscopy, cell microscopy, you can see normal duct, which has no shadow in between the circle. Then cancer, abnormal growth in the duct can be seen by some globules, some type of foamy material. It's like a foam inside. More foam, more particle you see like this, danger is there, more danger is there. If do not see anything inside, that means that's normal. That's a good one. We show next one. Similarly, ovarian cancer. You know, one thing I must tell you, that is I have seen in my life, when I discuss something about prostate cancer, colon cancer, I don't get that much support. But when I discuss breast cancer, ovarian cancer, I get more support. Ovarian cancer, like breast cancer at stage number 4, is very difficult to cure. But early detection can save life. And I will show you, I have already shown you the results against breast cancer and ovarian cancer. I will show you more. We show next. Here, you see how the structure activity, how the modification of the molecules can lead extremely potent anti-cancer drugs. Look here. Here, the molecules comes from casein, but the orientation of the groups are different. It is in the 2 and 8 position, and look the data. Cisplatin data, my molecule 1A, 1B is more active, much more active than cisplatin because it has lower number. Lower number mean using less amount of molecule, we can inhibit the cancer cell production. BRO melanoma, MCF breast, MDA breast, ovarian, SKOV ovarian, PC3 prostate, HL60 leukemia, leukemia, HT29. Now look, with respect to cisplatin, these two novel molecules have demonstrated significant anti-cancer activity. I have patented this to United States and world. I did not publish it because of patent yet. But starting from a molecule casein, we have seen tremendous activity. Now we see these type of molecules are active against colon cancer. Like before, early detection can help. We show next one. Here, we have shown here a colon cancer in vivo experiment, mean actual in the site, not in cell, actual in site. What I see here, you see the control experiment. After the addition of the molecule I have shown, one molecule is 1, we see a significant reduction of the tumor size from 450 mm to 216 mm after 3 weeks. So you see the reduction of the tumor volume after 27 days. We can reduce the tumor of colon, cancer tumor of colon by adding this molecule from 450 original diameter size, it became 216. And we see all the mice are active. The reason I said all the mice are active after 27 days, it means a lot, because that means the molecule could not generate toxicity. Many pharmaceutical scientists do not like to work on an agent if they find the molecule is toxic. I do not like to work on toxic molecule either. But at the same time, we need to see whether we can keep the animal alive. Since the molecule helped to destroy the cancer cell in the site, at the same time, mice have not died. The results demonstrated herein is very interesting and promising. We show next. Then NCI, National Cancer Institute of America, has selected my molecules to study in 60 human tumor lines. NCI, USA NCI has investigated my molecules against 60 human cell line, cancer cell line. They found our molecules are active, but they could not define the mechanism by which this molecule destroy the cancer cell. This is complicated work, I told you before. Can you show me the next? Now I am showing whatever I told you before. The cancer drugs act through DNA, RNA, and protein inhibition. Here, look the beautiful part, the molecule, active molecules at low concentration, which is 3 microgram, very low concentration, has no effect on DNA, has minimum effect of RNA, but lot of effect against protein. So what I told you before, when scientists believe when the molecule has aromatic system, they must act on DNA and RNA. That is not true. That is not true. Here, our molecule do not act on DNA and RNA significantly very little, but RNA protein inhibition is more. At low concentration, let's see at high concentration. We show next one, please. Here we see when I increase the concentration to 10 microgram or time, you see the protein inhibition goes to 100%. Still DNA, RNA inhibition, they rise, but protein inhibition, when you see the picture, you see the protein inhibition of the cell much more than DNA and RNA inhibition. This reflects that aromatic rings are not mandatory for DNA and RNA inhibition, which is significant study actually. Because when you see the molecules, aromatic molecule, everybody will claim they interact to DNA and RNA. All right, next on, please. This show next. Here, same thing, the trend, it differs 24 hours against leukemia cell. You see the DNA inhibition, RNA inhibition is there, but lower than protein inhibition. So when we change the cell line, we see the similar trend of the molecule. This study indicated the protein synthesis by the cell may be one of the most important mechanism, because what happens, cell cannot synthesize protein and eventually they die in presence of this molecule. They fail to synthesize protein. This indicates the importance of protein to cell also. That means a cell cannot make protein, that cell may not be able to function well. That's why they die. Next, please. Here, significant result after 48 hours. Look, DNA RNA inhibition is very low. Protein inhibition is much more. So in contrast to all existing speculation, we discovered some molecule that inhibit protein of the cell, cancer cell, but may not have effects on DNA and RNA. Even they have effect, that effect is very minimum. Next, please. Now I'm showing the surface of a cell by carrying electron microscopy in presence of our compound and in absence of our compound. Here, this cell, HL60 human leukemia cell structure in the under scanning electron microscopy looks like this. Now we show next one, please. Now in presence of the molecule, I have shown you a 2.5 micromolar compound. You see the surface of the leukemia cell has changed completely. What is the meaning? Meaning is some molecules can really affect this anti-cancer activity through membrane interaction, may not be interaction through DNA, RNA. This sort, if there is no interaction, the surface should not change. Now I am just changing the molecular structure to show the other types of aromatic system may have similar results. Next. Next. Here also similar molecule, different structure. Next. Here also I am showing different skeleton, different structure, and to improve the bioactivity, anti-cancer activity, to improve the pharmaceutical profile. Next. Next. You see diverse structures, diverse molecule can be prepared using fundamental knowledge, using exploratory knowledge, using the chemistry which is known, or developing new chemistry which is not feasible. So we can see, but when you identify molecules are good enough, then target would be to identify most potent drug and less toxic drugs. That's why our idea was to find out more potent and less toxic molecules. Next, next. Here I wanted to add some carbohydrate to these cancer drugs, because knowing the carbohydrate you need in cancer molecule can help the anti-cancer activity. Next. Here, look at the system. Here, the polyaromatic system is connected to lactam, which is antibiotics. The lactam unit is connected to sugar, connected to polycyclic system. And this lactam unit, penicillin or cephalosporin, thiamycin, augmentin, amoxicillin, these are all antibiotics. Now here I have beta-lactam unit connected to polycyclic system and connected to carbohydrate. All these three components can be knocked out easily by plasma degradation in body. And therefore, we can have different types of activity from three different components, which is carbohydrate, which is lactam, which is the polycyclic system. These kind of compounds are active against different kinds of cell. So in conclusion, what we have shown that we can make different types of polyaromatic molecules. Then we can add different types of heterochain, carbocyclic chain, heterocyclic chain. We can also add electron donating, electron withdrawing groups. We can change the aromatic core. We can add polar group. We can alter the functional group from amide to amine and amine to heteroamine, carbocyclic, heterocyclic. We can add natural product to increase the solubility. We can add peptide, we can add amino acid. We see the molecules act through DNA, RNA, or the cell membrane. Significant inhibition against protein has been discovered. Scanning electron microscopy was used. In vivo experiment was successful against a variety of cancer cell line. Mechanism of the action, how this molecule act against cancer cell have also been investigated. Next. This work was possible because of a number of people who worked with me in the project. And I have a big research project for the last many years. And as I told you before, the interdisciplinary work is very important, provided the person lead the program has good interpersonal skill and good problem-solving ability. With the help of good problem-solving ability and interaction with a number of professionals from different fields, one can really create a unique team, and this unique team can perform different types of interdisciplinary work. I am grateful to many of my collaborators who contributed in this project. However, I cannot write all the names of the people. Only some names are mentioned here. And you see, they have done a lot of work with me, and they have been doing a lot of work with me. I am recruiting people all over. I am trying to identify good collaborator, honest collaborator throughout the world. And next one, Bishop. You see that this grant was possible, this work was possible because of support. It's a competitive program, and this was supported from US government, but through multiple grants. And I am very grateful to them to understand our research program. And I am grateful to all of the student, faculty members, my friends, family, and the future friends, current friends. I have many, many very good friends in my life. I am very fortunate that to have you many, many friends. And finally, I thank you for organizing this program. And I hope in the future you will do the same, and you will continue to do the same. And many scientists will be able to initiate different types of interdisciplinary program, and this will help you faculty development program and also many students in the future. Thank you very much for your kind help and attention. Thank you. I'm sorry we had some problem in the beginning, and always problem starts. To solve the problem is our goal. So never be frustrated and work hard. Move out from your comfort zone. The zone you feel very good may not be good in the future. That's why always to produce something, you have to go out from your moving zone, from your comfort zone. You face the difficulty and challenge. Thank you, sir, for your attention. I'll be happy to answer you any questions that you may have. Thank you, sir. Thank you, ma'am.
Thank you. you, sir, for your nice presentation. So we have not received any questions on the chat box. So I think they have no doubts regarding this research activities what you have performed and cleared all these things.
Thank you so much, Professor Banik sir, for your valuable presentation. We remain most grateful for the rapt attention with which you have delivered this talk. May I now request Principal Professor B.V.V. Ravikumar sir, coordinator of this event, to present the certificate of appreciation to Professor Vimal Krishna Banik sir as a token of our love and affection from entire RIPS fraternity.
Thank you, Professor Banik sir, for sparing your valuable time. And I must congratulate and appreciate you for all the good work you are doing to the society through your research. Please accept this certificate of appreciation on behalf of the entire family of Roland Institute of Pharmaceutical Sciences. Looking forward to meet you through webinars or, if possible, physically. Thank you very much, sir, for coming to RIP's platform. Please accept this certificate of appreciation.
Thank you very much. Thank you so much.
Thank you, sir.
Namaste.
Thank you, sir. Namaste, sir. With this, the session 1 of Day 1 Series 2 FDP program comes to an end. We will be resuming the session 2 of the program very soon on the same platform. Good afternoon and welcome back to the Day 1 Series 2 and Session 2 of the online FTP program sponsored by AICTE. We have with us our next resource person, Dr. Sarwar Beg, with us. Before starting his eminent talk, let me first of all introduce Dr. Beg to all of us. Dr. Beg has completed his PhD from Punjab University, Chandigarh, and his M.Pharm degree in pharmaceutics from Jamia Himdard University, New Delhi. Dr. Beg is now currently serving as Assistant Professor at Department of Pharmaceutics, School of Pharmaceutical Education and Research, Jamia Hamdard University, New Delhi, India. Prior to joining Hamdard, he was working at Jubilant Generics Limited, Noida, where he was solely responsible for implementing the QBD framework for generics and new product development for regulated markets like US, EU, UK, and contributed in ANDA filing of more than 25 generic products. Dr. Beg has over a decade of experience in systematic product development and characterization of novel and nanostructured drug delivery systems using QBD and computational approaches like design of experiment and multivariate chemometric techniques. Dr. Beg has authored over 180 publications in various peer-reviewed journals, which is having a cumulative impact factor of 430, H-index of 34, and 4,500 citations. Also, he is having 60 book chapters, 17 books, 13 journal special issues, 3 Indian patent applications, and a score of conference presentations to his credit. Dr. Beg has received several Young Scientist Awards at various national and international forums, including CIPRA Innovative Pharma Research Award 2016 and Bronze Medal, Udrazit Award 2015, Budding QBD and ADMET Scientist Award 2014 and 15, Novartis Biotechnology Leadership Camp Award 2012, and a score of Best Paper Awards to his credit. In 2017, Sir was felicitated by Honorable Health Minister of India and Managing Director, Sun Pharmaceutical Industries, with Sun Pharma Science Formulation Young Science Scholar 2017. In 2019, Sir has selected for UGC Research Award with 10 lakh rupees startup research grant. Very recently, Dr. Beg has been awarded with prestigious AMS DBT Newton International Fellowship to 2020 as an early career researcher to pursue research in UK with a grant value of 15 1 lakh 50,000 US pound. Besides, Dr. Sarwar is also a recipient of National Postdoctoral Fellowship in 2017. Sir has also qualified GATE 2018 with the All India rank of 38. With this brief introduction, let us now welcome our resource person, Dr. Sarwar Beg, on board.
Thank you, Dr. Rajaram, for the nice introduction. I'm privileged, I'm happy, and I'm fortunate to again have an interaction with all of you. And to the all faculty members of Roland, I am part of Roland Institution. In 2008, 2010 to 11, I was with Roland. So it is a part and fortunate privilege that I am again back to you guys to have some discussion about the concept of quality by design, more update on regulatory aspects. So good afternoon, Principal Sir. Good afternoon, Niranjan Sir. I hope all of you are well, and I am very happy that Roland this time has arranged this prestigious AICTE sponsored FDP, because I have seen so many institutions, universities in North India basically they are arranging such a AICTE sponsored FDP program. So it is a happy moment for me that yes, Roland is arranging such kind of program for this particular eastern region and in the region of Odisha and Andhra. So many faculty members will get benefited over this program. So I'm back to presentation. So just a second.
Share your screen.
Dr. Beg,
Yeah.
share your screen.
So I hope it is visible to everyone. Is it visible to everyone? Yes, it is visible to. Yes, yes, yes. I will make it full screen. So is it visible now, full screen?
No, sir, you make it full fast.
Okay. I need to. So
Yes,
is it
yes.
fine now?
Yes, sir.
So it is
You can
fine now.
continue.
Yeah. So at the onset, the topic of interest is the systematic approach of quality by design. I have chosen, which is very close to my heart. Where basically I have worked almost 8 to 10 years on this concept, and it is a matter of pride and privilege that I can present some of my recent experiences and some of the related updates in the field. Most of us might have already heard many times about the implementation of QBD and how quality by design is basically playing a great role in the pharmaceutical industry, and of course in academia also the requirement of pharmaceutical quality by design is high, be it the any field of pharmaceutical development like the formulation development, the analytical development, the chemical synthesis, the extraction process. So all field of pharmaceutical research has a close link with the application of pharmaceutical quality by design. So my presentation has been structured into several parts. Uh, the quality scenario in pharmaceutical area, biopharmaceutical area, what are quality by design various paradigms. So disruptive pharmaceutical technologies that the QBD basically embarks upon, the 5 steps QBD approach, and there are various QBD models and experimental designs. So application of quality by design for formulation development, and there are some holistic development with respect to the case studies. So altogether, I will discuss a couple of things which are related to the case studies also, and there are some QBD cautions and conclusions to you. So going back to a news, very famous news article, some of you might have gone across through this news, which was published very lately in 2016. So generic drugs are basically the most highly sought after medicines, as we know that in many of the developed world and developing world also, the generic drugs are considered to be very the cheaper alternative over the branded medicine. So Indian medicine, the news article punchline was the Indian medicine available at 1 of 100 cost, which saves the life of an Australian patient. So the drug was sofosbuvir, and it was marketed in the name of Sovaldi. So Sovaldi was the brand name of the drug. So the patient, whenever he was prescribed in Australia about the branded medicine, the cost was nearly about $100,000. But certainly when the patient looked for the alternative cheaper options, so the generic medicines which were available in Indian manufacturer, so they were nearly about $1,000, which was saving around the 1 of 100 cost of the patient who was prescribed with this particular drug. So the crux of this point is like the generic drugs are considered to be very safer, and they are very cheaper. However, the quality of generic drug is a big concern these days, since so many pharmaceutical industries are facing a regulatory concern with respect to the product recalls and product rejects due to the inconsistency in quality, to the poor quality of the generic medicines. So there is a point of concern by every other regulatory agencies around the globe. So very famous slide. So during the time of Obama era and before that also, the American market, which is considered as the most potential market for the generic drug filing. So they have already identified that time that generic drugs are considered the cheaper options. And so every year the generic medicine save the patients and US healthcare system of around $160 billion, or in Indian rupees it will be nearly around the 11 trillion rupees, Indian rupees, the total size of the market of generic products. So generic drug approval, how does it means to FDA, and how does it means to us as the pharmaceutical scientists, and how does it means to the regulatory authorities? So it is a different scenario. So everyone perspective has been different. So FDA says that think it is easy becoming a generic drug in America. So think it again. So the point of discussion here is to become to register a generic drug in the US market with the FDA, US FDA as the key regulatory agency. So it has become very difficult these days to get assured the quality of the generic product, the content uniformity, the labeling requirement, the rigorous manufacturing standards that the industry need to follow, the pharmaceutical process validation data that the industry need to show. That also is a requirement with respect to the generic drug application filing is concerned with FDA and several other the federal regulatory agencies. So to make sure your generic drug meets approval, FDA says that it should meet your approval first. It should meet your understanding first in order to meet our understanding. This is the quote of FDA. So as far as the generic products are concerned, they are basically the pharmaceutical clones. So generic product is a comparable to a branded product in all quality characteristics except all quality characteristics, and primarily the bioavailability requirement. The bioavailability of generic product should be equivalent so that we can achieve the bioequivalent products with that of the reference product. So the key requirement is the purity, quality, strength, performance characteristics, and the bioavailability. So biopharmaceutical products, if you compare, these are the biological products or biological medicines we call, and the copy form of the biological medicine or biopharmaceutical are called as biosimilars. So now the era of biosimilar has been come into picture. The generic era of biological product, which is quickly taking place with respect to the filing of the biosimilar applications too. So biosimilars are the cost-effective alternative of the biological products, basically the vaccines, basically the sera, basically the hormonal products. So these are called as biosimilars. So biopharmaceutical market again is booming very high with respect to the cost of the development is concerned vis-a-vis the branded biological products. So altogether the scenario is very gloomy with respect to the generic medicines are concerned or biosimilars are concerned. So a pharmaceutical drug delivery system, basically, I will continue more about the pharmaceutical drug delivery system. So a pharmaceutical delivery system is basically an intricate device which contains mixture of the API, the drug substance, the polymers, the excipient, the surfactant, the emulsions, the suspending agents. So many things are present in a typical formulation that we have seen. So altogether, every constituent of the formulation may be responsible for final quality characteristics of a drug product. So the holistic essence of achieving a robust drug product involves identifying the nature of individual constituents of a formulation or a product, which can directly or indirectly influence the quality attributes. So a difficult generic, the term that right now these days it is going on, the difficult generic or complex generics. So the complex generics are nothing but the specialized pharmaceutical products which are having the release tampered, or the release has been changed, the control release product, the modified release product, or the products which are specifically delivered to a particular site, like the liposomal product, the nanoparticle, the conjugated nanoparticle system. The generic form of nanosystems are also right now available in the market. So a broad range of classification of complex generic involve the conventional product as well as the specialized product, which involve both the modified release system as well as the complex liposomal nanoparticle systems, which are altogether considered as the complex generic products. So generics are basically India is the pharmacy capital of the world, and India is also considered as a generic hub of the world. So manufacturing of generic drugs which are maximally sent to outer market, to the outside world, are basically made in India. So 40% generic drugs sold in the US are from India, and every third DMF, drug master file, for approving authorization of the drug substance manufacturing in US are from India. So this is a huge $1 billion huge market, and it is rising from 22% rate of from 2020. This is the old data that I have. So the latest data may be higher than 22% rate, and that's how we expect that generic drug market is flourishing at a very high rate. So the key point is that whether the generic products are safe and how the generic products are being accepted by the regulatory agencies and the end consumer. So a survey indicates that there is a skyrocketing increase in the number of product recalls and rejects in the last couple of years. If you consider the 10-year data from the FDA side, if we look at the 10 years data of market recalls and rejects in generic product, then it is very high. And if we go for the number of filing of generic product each year, that is also very exponentially increasing. So the filing is very high, the recalls is also very high. So which indicates that the products are of not adequate standard that are expected by the regulatory agencies. So poor quality is a major issue with respect to generic product. So we can look at the graph, how the generic product market is distributed. So how the generics are basically distributed. So the blue color segment indicates the prescription generic products, while other segment indicate OTC cosmetic products and medical devices, dietary supplements. There are also generic players in those segments also. So mostly we are concerned about the prescription drugs because they are therapeutic. They are under the therapeutic segment. So we can see that the nature of generic product market is drastically changing, and this particular slide indicates that why we are more concerned about the quality of generic product. Say we have 10 number of generic product for a particular drug in market, and the FDA says that the bioequivalence criteria must be met with respect to 3 important requirements: prescribability, switchability, and interchangeability. So what is prescribability? So a physician write a prescription, if physician write a drug to the patient, for example, a paracetamol 500 mg tablet. So if the patient can go to the chemist and he asks for the available products, available brands of paracetamol tablet 500 mg strength, so the chemist can offer 3 to 4 brands or tens of brands to the patient, but it is the choice of patient who is basically looking after the cheapest medicine that is available on that particular drug. So it's all about the switchability. It's all about the prescribability. There should be a flexibility with respect to the prescribability. There should be a flexibility with respect to the switchability. When there will be the bioequivalency, the switchability of one brand to another brand one generic to another generic is very, very easy. That's what the FDA wants. That's what the FDA is looking for, that all generic products which are available in the market and the reference product available in the market for a particular drug must have identical strength, purity, and bioavailability criteria, so that the patient is free to switch any product despite of despite irrespective of the quality, irrespective of the purity, irrespective of the bioavailability criteria. So prescribability, switchability, and of course the third parameter which is not highlighted on this particular slide is the interchangeability. So product can be interchangeable. The consumer, the end consumer, the end patient must have full freedom to interchange with the product. We change the generic product based upon the cost criteria, based upon the feasibility of his pocket. So altogether, FDA wants that the quality should be ensured, the bioavailability should be ensured, so that the freedom of the patient will be maintained, freedom of the end consumer, which is the patient, should be maintained. So high product variability is the major issue with respect to the quality. So variability is a term which is described in terms of the quality consistency. When the quality of the end product is very high, the quality consistency from batch to batch manufacturing is very high, then certainly we expect that the variability is very low. Variability with respect to the quality is very low. So high variability in quality is a major concern in pharma drug development, pharma product development scenario, and federal regulatory agencies are forcing the pharmaceutical industries for reducing variability in their product to maintain consistency in batch to batch fashion. So you can see the graph. We can see the graph. There is a product, branded product, and there is a generic product. For branded product, we can see there is the graph of batch to batch production cycle is not very zigzag, and it is showing a smooth pattern, except some deviation in some of the batches. But the normal trend is showing that the batch to batch quality is within the confidence intervals of acceptability, within the confidence intervals of the consumer acceptability, within the confidence of the regulatory acceptability. But in case of generic product application, you can see there is a heavier trend, there is a quick zigzag pattern, and how the variability is showing that the high variability is showing that the trend of the quality of achieving for a particular batch is going beyond the threshold limit of confidence interval. So altogether, the quality is major issue, and the quality is basically and that's what the regulatory agencies are basically looking forward. So the major reason behind the poor quality in the pharmaceutical generic products and the pharmaceutical drug product manufacturing from decades is due to the reason that the pharmaceutical industry is engaged in producing Six Sigma products. The pharmaceutical industry is expected to produce products having Six Sigma standards, but the manufacturing techniques are of Three Sigma standard. So what is Six Sigma and Three Sigma? These are basically the concept which are derived from statistic of the number of defects, basically the parts per million of defect that are observed with respect to the number of products which are manufactured during the production cycle. So the Three Sigma process indicates that the capability of the manufacturing processes that we are using with respect to the automation is required, with respect to the artificial intelligence is required, are very low. But we are expecting somewhat the standard should be of Six Sigma level, that our defects should be very low, which is very unpractical scenario these days the pharmaceutical industries are facing. So the current scenario is we should achieve a Six Sigma quality of the pharmaceutical product, but the instrument, the pharmaceutical processes, and the manufacturing standards that are used should be of Six Sigma capability. So this is how the Six Sigma is explained. The current state is Three Sigma, and the desired state is Six Sigma. And Six Sigma indicates that the defects are very less. That maybe like three to four defects per million of the products which are manufactured in the industrial production cycle. Whereas in case of Three Sigma, the defects can goes up to 66,000 defects per million of the products which are manufactured. So we can see that the defects are very high as far as the expectations are concerned. So altogether, the answer lies in why the variability is coming and how the variability is coming in the production production or pharmaceutical development. So we know that the pharmaceutical product development contains a series of processes. So it contains a series of processes starting from the. This is the example of a. This is the example of immediate release tablet manufacturing, where the raw materials are converted into the finished products, where the granulation, the coating, compression, milling, compaction, and finally the film coating process takes place to convert the raw material into the finished products. So understanding the variability is very high, very important, and the variability can be comes from the sources like the API, the excipient, the processing material, the processing methods, or the manufacturing methods, which may influence um, the pharmaceutical product variability, quality variability. So variability is the key issue, and how will variability can be reduced? So variability can be reduced by fixing the processes, manufacturing processes that we are used. So we have variable raw material, whatever the drug substance, whatever the excipient that we take from the manufacturers or the suppliers of the API and excipients contains a very high variability. So we cannot certainly reduce the variability with respect to the raw material that are provided by the supplier. Despite we ask them for a stringent specification, we still the raw materials which are provided by the suppliers faces high variability at times with respect to the particle size of the drug substance, with respect to the particle size of the excipients, physicochemical parameters of the API and excipients that we receive from the suppliers are quite a very high. So we have to certainly reduce the variability which is encountered during the process. So we have to optimize the process. So the optimization of process can lead to the robust product with low variability in the end product quality. So inapt amalgamation at times can lead to weird results. So hence systematic pharma product development is a must. You can see the scenario how the weird situation has been developed. So the systematic pharmaceutical product development is a key criteria that can reduce the quality crisis, that can reduce the issue with respect to the poor quality. So generic product, how can quality be assured? By reducing the variability through optimization. So altogether, we have to reduce the variability by implementing suitable optimization techniques. So there is a famous article which was published in Wall Street in Forbes magazine in the 1996, and the Forbes magazine is very famous and popular to all of us. People generally look into Forbes list for who are the 100 most powerful people, or who are the top 100 richest people in the world. But this particular magazine basically quoted an article that new mantra of multivariate testing. So if you test all the factors one at a time, there is a very low probability to hit the right one before everybody gets sick and quits it. So the point of discussion of this particular quote over here is the pharmaceutical product development includes so many factors. So many process parameters, so many product related parameters are involved during pharmaceutical development. So if one is interested in optimizing each factor at one time, so one factor at a one time approach or one variable at a one time approach can lead to no significant conclusion with respect to the issue with respect to the quality is concerned. So the ideal scenario could be the application of the multivariate testing, where all the factors can be simultaneously considered for reducing the variability. Another famous article which was quoted already in 2002, the Wall Street Journal indicates that the entire pharmaceutical industry is facing quality crisis, but the other manufacturing industries like the potato chip makers, the pizza industry, dental implant industry, oil exploration industry are far away from the quality standard that the pharmaceutical industries are using. So this was a big slap to the regulatory authorities, basically the US FDA, and that exactly changed the scenario of pharmaceutical development. So FDA described the concept of variability, the concept of product quality in 5M approach. So this approach is called 5M approach, where man, machine, milieu, material, methods, measurement, and management are responsible for overall quality of a product, overall quality of a drug product. So this particular diagram is called as a Ishikawa fishbone diagram, and basically the variability which is coming from the men, which is coming from the formulation scientist, which is coming from the analytical scientist, which is coming from the machines which we are using, and which are and basically the experimental methods which we are using, the variable raw material which we are using. So all together, the variability coming from various sources that a pharmaceutical product pass through can influence the end product quality. So somehow we need to churn out the variables or with the factors which are actually possessing, which are actually incorporating variability in the end product. So pharmaceutical quality, there is a need and demand. The regulatory need is to ensure the quality. The company repute is to ensure the quality. The patient also requires a quality product. The market rivalry is also there. Whenever two companies, whenever there are tens of players in a particular product, and someone is producing a substandard product and someone is producing a product having quality at par, the consumer demand, the market rivalry exists, and the consumer acceptability for a particular brand goes high. So that's how the scenario takes place with respect to the business world, and ultimately the patient satisfaction with respect to getting a consistent quality product for therapeutic purpose is also the need of the hour. So pharma product development quality scenario, the quality at high cost and effort, and inefficient, insufficient design can lead to poor quality, and that's how the FDA is expecting that the pharmaceutical industry need to change the manufacturing standards. And the famous quote by the FDA director Janet Woodcock indicates that. So she has explained the pharmaceutical quality. She has defined the pharmaceutical quality as a function of the quality attributed from the drug substance, the quality attributed from the excipient, the quality attributed from the processes and packaging materials. So altogether, the pharmaceutical quality is a function of various variables which are attributed from the drug substance, the packaging material, the excipients, and processing parameters which are required for it. So quality needs planning. If you do not know where you are exactly heading towards or where moving towards, so you will get lost in somewhere else without getting the right conclusion. So planning of quality from the very first end is the actual need of the hour. So, and the federal agencies like FDA are basically constantly pressing the pharmaceutical industry to adopt the systematic approaches, and quality by design is one of the systematic approaches that can lead to consistent quality of the drug product. So as per ICH definition, the ICH defines quality by design as systematic approach for development that begins with predefined objectives and emphasizes on product and process understanding based upon sound science and quality risk management. So ICH, who instituted the guideline ICH Q8, Q9, and Q10 guideline for development of pharmaceutical products. So ICH said that the quality by design should be started with very first stage of the product development, and the manufacturer must define the objectives from the very first end. So these are called, that's why they are called as predefined objectives, and emphasis should be more on product and process understanding. So understanding is very important. Doing thing is not important. Doing thing is important for us, but whether we are doing by understanding this scenario, understanding the things, that is more important for us as well as the regulatory agencies, because the problem of quality crisis actually lies in poor understanding. We do not understand much into the process variability. We do not much understand much into the variability which is coming from raw material. So we do develop the products, we do develop the pharmaceutical products as a formulation scientist, but we do ignore the variability that are associated from various sources. So there was a famous quote that there was a famous understanding that I got from a very closely associated person who is working right now these days at a as a leader of one of the pharmaceutical industry, that including a chemical engineer, including a process engineer in pharmaceutical development can really solve the quality crisis. So this is a very important and interesting thing that we are learning the process development, we are learning the unit operation in pharmaceutical education, in B Pharm education, in M Pharm education. We teach student the subject unit operation, but hardly we bother about the importance of unit operation. We teach industrial pharmacy, but hardly we take the right importance of the statistics in the industrial pharmacy, the industrial the manufacturing development. So those things are very important with respect to the ensurement, with respect to the ensuring the quality is important. So what is quality, what is design, and how can design deliver the quality? That is the main definition that I feel to describe over here. So people ask me what is quality and what is design, and how can design can deliver the quality. So that's all about the quality by design. So what is quality is a very simple thing, that if you look into the meaning of quality in Oxford Dictionary, you will find that building excellence in a particular trait is called quality. And what is excellence? Excellence is nothing but building quality in a particular trait. Industrial trait is called excellence. So quality and excellence are interchangeable, and quality is a dynamic. So many scientists have defined the quality in their own way. So what is the definition of design? So design is nothing but the systematic and conscious intent of doing things right from the beginning, with predefined quality mindset to satisfy the consumer need, is called design. So design can deliver the quality based upon the end product objectives, the end goal objectives. So ISO also define quality, the degree to which inherent characteristics of fulfilling the consumer requirements can also be the definition of quality by ISO. Dictionary.com has its own definition. Quality is perceptional, conditional, and selective attribute understood differently by different people. And altogether, quality is dynamic. Quality is not static, and quality is dynamic. The quality is continuously changing, and we have to evolve with time, with respect to the consumer demand, with respect to the patient requirement, with respect to the quality products are concerned. So pharmaceutical quality needs and demands, ICH-Q6 guideline gives this quote that ability of a product to satisfy the stated needs of consumer, including the identity, strength, purity, without any undesirable side effects, is the need of the ISH, and that's what the need of regulatory agencies too. So quality with respect to the regulatory demand and consumer demand, it should be fulfilled at every stage of product development. So the concept of quality by design came into picture very late in 1991. The concept of quality by design was defined by James Juran, but the story of quality and how the quality term was built up. So the term quality was described by W.E. Deming in 1980s, and before W.E. Deming, Philip Cosby, he gave the concept of the culture of quality. So the concept of culture of quality was started in 1950s. So after 1950s and 1980, Motorola gave the concept of Six Sigma, and Motorola started implementing the concept of culture of quality. And in 1990, Juran gave the concept of quality by design. So Juran gave the quote that quality could be planned in the very first place to avoid quality crisis. This quote has been several times used by several news articles, several research articles, review articles in the domain of pharmaceutical quality by design. But this quote has real significance these days also. The quality could be planned in the very first place to avoid quality crisis. So the pharmaceutical industry, which are currently facing with, issues with respect to the product recalls, with respect to the poor quality, with respect to the product rejects, with respect to the poor quality. So the reason behind is the lack of understanding and lack of planning from the very beginning. So when will be the plan? When the planning will be there from beginning regarding the end product objectives with some predefined measures, then there will not be any issue with respect to the product recalls and rejects. So Juran's philosophy says that quality is by planning. Quality cannot be achieved by chance or by luck. So it is all about quality by planning. So it needs hardcore planning to ensure the requirement with respect to the end product objectives. So some people asked about what exactly the quality by design is. Some consider it as a software, some consider it as a regulatory edit. Is it a program or is it like a software? So exactly the quality by design is not anything which is written on this particular slide in the form of question. But what exactly the quality by design is? The quality by design is the philosophy. So it is a philosophy. It is not any software. The QBD is not any software. The QBD is not any tool, but rather it is considered as, it should be considered as a philosophical approach, philosophical thinking, philosophical rational mindset of doing things right from the beginning, with predefined objective, with predefined measures, with predefined set of procedures. And of course we need to conglomerate, we need to incorporate scientific principle, we need to incorporate scientific principle in the development, in the product development. And we have to bank upon certain statistical criteria, which we need to bank upon certain statistical parameters, statistical evaluation approaches like the design of experiment, multivariate statistical techniques. Then only the end product objective of getting the quality consistency can be satisfied. So doing it right from the beginning is the objective of QBD, and it's all about the patient, and we are more conscious about patients. So understanding the patient need is important. Designing and developing products meeting the patient needs, it again is important, and we are always concerned about the end consumer, the end patient in the mind. So quality cannot be tested into the product, it has to be built into the product by design. So quality and patient are the inter requirement, and the requirement needs the end consumer, the patient requirement need to be satisfied. And good pharmaceutical quality represents an acceptably low risk of failing to attain the desired clinical attributes. That's what the FDA also requires to be made with respect to the pharmaceutical product manufacturing. So besides the advantages of getting the consumer satisfaction, the quality by design approach is also important for improving the product robustness, to ensure the enhanced knowledge sharing. It also ensures the time, it reduces the time to reach the market, it reduces the recalls and rejects that I have highlighted several times. So it also reduces the generic skepticism that I have, as I have mentioned in my previous slides also, that due to the poor quality, people to doubt about the quality of generic product, people to doubt about the safety of generic products. So that safety concern or skepticism in getting the desired therapeutic needs from a generic product can be removed by use of implementation approach. And the quality by design can reduce the minimum post approval changes that the company need to require after filing the generic product application into a regulated market like in US or Europe. So ensuring the quality is very important. So the understanding of quality by design need to be very carefully done. So initially it will take time for the understanding of the integrity of the concept of QBD. So it will take, the current state is like increasing resource, the time of learning the key points about the quality by design approach will be very high. The development cost for training the personnel who are involved in development will be very high. So initially the current state will be the investment phase, where the companies need to train their scientists, the company need to train their people to ensure that the learning of quality by design should be there from the very first end. Then only the desired state will be a cost effective approach. More knowledge sharing will be done, more flexible and continuous improvement in the development will be achieved. So that's what the regulatory agencies do expect. So phenomenal cost benefit ratio is there with respect to the understanding the concept of quality by design, and the later stage acceptability of the product, later stage of getting the product with right quality will be improved. So we can see that from this particular slide, the scenario of the post approval submission load. So this is the left side pile indicates that the 2003 submission of the dossier, which is very hefty dossier, and the right side dossier indicates the 2006 onward, after the quality by design approach was instituted and the ICH guideline was published in 2004. ICH Q8 guideline was published in 2004, and the revaluation was question based review. So with the QBD around, the QBR was also implemented. So initially the QBD was implemented, and with QBD around, the QBR was implemented. So QBR is called quality question-based review. So QBR is a question-based review approach that FDA, after filing of the generic product application, tend to ask the pharmaceutical industries to answer certain queries whenever there is poor understanding about the particular submitted application. So that is the whole scenario is called, the whole process is called question-based review. The direct review and discussion comments between the regulatory agencies and pharmaceutical industry is guided by a process called question-based review. So to design and develop the drug products with the desired attributes, so quality by design is the ultimate tool, and it ensures quality excellence, it ensures customer satisfaction, it provides ease of production. So ICH Q8 guideline, Q9, Q10, 11, and right now the Q12, Q13, and Q14 guidelines are also coming in the next few years. Already Q14 guideline draft has already been published by ICH, and the FDA US FDA has indicated the suitability of this guideline, that guideline for the generic and the new product manufacturer. So the Q12 guideline is basically the guideline which is focusing altogether on product lifecycle management, where the Q8, Q9, and Q10 guideline can be used in consonance to understand more about the pharmaceutical quality system and pharmaceutical development. So earlier Q8, 9, 10 were proposed, Q10 and Q11 came, and Q12 was also came in 2014, and now Q13 and Q14 guidelines are also coming. So guidelines are very important for the pharmaceutical industry as far as the regulatory demands and regulatory standards are concerned. So guidelines are basically the guiding tools. So guidelines are not mandatory requirement. Guidelines are basically the requirement that regulatory agencies tend to encourage the pharmaceutical industry. So Q8, 9, 10, 11, 12, and right now 13 and 14, which are coming, are being endorsed by majority of the regulatory agencies all over the world, be it FDA, MHRA, MCC, WHO, the China FDA, the Saudi Arabian Authority, the Anvisa Brazil, PMDA Japan. So majority of the regulatory agencies altogether have endorsed, and even the Indian regulatory agency, the CDSCO, is on the way for drafting a guideline for quality manufacturing, or they are in the way to draft a guideline on pharmaceutical quality management system to ensure the quality of the drug products manufacturing within the country, within this country. So FDA started implementing the concept of QBD in 2010, and 2013 was given as the deadline, and 13 has already been passed almost 8 years back, and we are still discussing the concept of QBD, and the people are still in the way to understand the actual scenario of the expectation of the regulatory agencies as far as the concept of QBD is concerned. So situation is still alarming, but we need to act very cautiously. Why? The understanding need to be done in a proper sense in order to achieve full implementation of the concept of quality by design in the industrial level. So quality by testing is a word that was earlier used when QBD was not there in place. So quality by design approach of manufacturing, quality by testing approach of manufacturing basically requires rigorous testing at every stages of product development, and the final end testing to ensure that whether the product is going right or wrong, whether the quality of the end product can be acceptable or not, by in-process quality control tests. So those who have worked in the production also, they might have seen that the production batches are passed through in-process quality control tests. IPQC tests are there. So those tests are very rigorous, very stringent, are made by quality assurance and quality control teams at production level, and that requires end product testing as well as individual in-process quality control tests to ensure the quality of the end products. That was the scenario is in with the help of the quality by testing approach. But when the quality by design approach came, the end product testing was nearly obliterated. Why? Because the the QBD approach basically highlights to work with predefined objectives. So when we are working with predetermined objectives with critical understanding on each stages of product development, the role of the end product testing nearly comes to the negligible level. So the product control strategy and the process control strategy plays a great role. So every stage of development is adhered to with the help of a suitable control strategy. So the control strategy provides continuous improvement. That is what the quality by design aims for. That whenever we are implementing QBD concept, there should be continuous learning. So one cannot be a master for any concept from the very first go. If I am manufacturing a transdermal patch formulation, I cannot be a master, I cannot be expert on very first stage of development, but I need to learn through the evaluation of continuous stages of development to continuous process of learning. That's what the QBD aims for. So traditional QBT approach, quality by testing approach, indicated that design is not very intensive. Design was not given very high relevance. So testing was dominant. So we need to ensure we were ensuring the quality by rigorous testing, which was cost intensive as well as time intensive, and many times the failures are reported at the very last stage of product development in the final stages of commercial level. So that was very cost associated approach. But when the QBD approach has come into picture, so design and testing were balancing each other. So every stage the use of systematic design approach was parallel with the testing approach. So that's how the regulatory agencies are expecting also. So this is a cartoon basically indicating that the man who is passing, who is a trespasser, who is passing through a building, having a long tower, QBT ivory tower. The man was asking the person, I'm afraid the building which is next door might be undermining your foundation, because the QBT approach is so testing intensive approach that it may not give you right ensuring of the quality from the very first end. While the QBD approach provides the assurance for getting the right quality from the very first stage. So application of QBD is omnipresent. So this is in nutshell. I can explain that the quality by design is an omnipresent approach. It can be implemented at any stage of product development, API development, analytical, and formulation. Beyond formulation in dissolution testing, manufacturing, bioequivalence in clinical trials, stability testing. So many advantages of quality by design have been come into picture. So QBD is now not a formulation intensive approach. It is not an analytical intensive approach. So QBD is present at all stages of product development. So if we look at the entire product life cycle, then QBD has role at each stages of product development. So 5-step methodology of QBD implementation. So the approach of QBD implementation can be done in a 5-step methodology. So step 1 involves defining the objective. So sometimes the technical terms, the objective is also called as QTPP, quality target product profile. So we need to enlist the important elements of the target product profile, and we have to identify critical quality attributes in the very first step of QBD implementation. In the step 2, the critical material attributes and critical process parameters are identified using a suitable screening experimental design, or we can avoid screening experimental design. We can bank on the risk assessment part in order to ensure the quality, in order to ensure the right selection of the variables. So step 3 involves the experimental analysis with the help of implementation of DOE, where actual role of experimental designs comes, and the factors which are identified as the critical or influential factor during the step 2 of the product development cycle are optimized in step 3, and step 4 involves modelization and validation of design and control spaces. So, and the step 5 is the pilot plant scale up, validation and commercialization at the production level. So that's what the 5-step approach of QBD is all about. So defining the QTPP is very important, which is also called as quality target product profile, and it is based upon the target product quality criteria basically. So what kind of product we are manufacturing, what is the need of that product, and what should be the drug release profile, what should be the pharmacokinetic profile of that product. So those things comes under the quality target product profile. And critical quality attributes are nothing but the key quality parameters that we need to ensure at the end of this the product development cycle, so that the quality of a product can be critically monitored. So QTPP example includes route of administration, dosage form, type, strength of the product, then container and closure system. All these things included in the quality target product profile. Even the resolution criteria, the pharmacokinetic criteria, and even stability testing and purity of the pharmaceutical product comes under the quality target product profile. While CQAs includes the critical quality attributes. So here is the example for the QTPP for a tablet dosage form. Dosage form, the route of administration, strength, tablet weight, appearance, identity, purity. These are the elements of the QTPP. However, some of the parameters which are derived from QTPP are taken as the critical quality attribute based upon their direct linking with the clinical safety of the formulation, or the safety of the formulation, effectiveness of the formulation, and safety of the formulation. Like the assay, content uniformity, and dissolutions comes under the effectiveness of the dosage form, and the impurity level, the impurity level or the amount of impurity which is present in a product comes under the safety level. So many a times there are genotoxic impurities, there are the carcinogenic impurities are there. So FDA tend to ask the manufacturing company to reduce the levels of the known impurities and unknown impurities as per the pharmacopoeial standard. That's what the actual scenario of identifying the QTPP and CQA for a particular product takes place. So step 2 is identification of critical material attributes and critical formulation parameter or critical process parameter. That's done with the help of screening. There is a principle that is called Pareto principle. So where the 80-20 ratio of identifying the product and the quality attributes which are associated with the product are governed. So the Pareto principle says that 80% of results are due to the 20% of effects, or in a simpler terms, the 80% of effects are due to the 20% of causing factors. So we need to identify only those 20% of factors which are associated with the product, and those 20% factors need to be optimized to get 80% assurance about the quality of the end product. So how the process of prioritization takes place? So whenever we have many X variable or many input independent variable which are related more or less related with the API, excipients, processes which are involved in manufacturing of a product, and many Y variable which are involved the quality criteria, quality attributes which are related to the end product, or even the intermediate steps of the product. So those things can be screened with the help of prioritization based upon the prior knowledge and prior experience. These products can be these variables can be identified. So factor screening exercise can be used. Use of experimental design is highly recommended during the factor screening purpose. So screening experimental designs are used. There are so many examples of screening experimental design. I will tell in the coming slides. So screening ensures identification of right variables which are actually impactful, or which are actually impacting the quality of the end product. So this cartoon basically indicates that the prioritization helps in extracting the unwanted outcomes, unwanted factors we need to identify, remove out, and we have to basically focus more on the factors and the variables which are important to us. So this particular dentist who is asking the particular dentist asking that your teeth look great, but it looks like we need to extract your tongue first. This should not be the approach that the dentist is basically focusing on. We need to extract the right variable at right time, so that the optimization exercise which is done with the help of experimental design should be fruitful. So step 3 is design guided experimental analysis using the experimental design. So step 3 requires the use of experimental design based systematic optimization, where various screening and response surface design can be used for optimizing the product parameters and process parameters for their net influence on the quality attributes of the drug product. So systematic QBD implementation requires role of quality risk management, screening exercise, and DOE implementation to ensure the right product quality. So what is design of experiment? By this time, all of you must have come across this term, the experimental design. So experimental designs are the systematized approach. They are mathematical and statistical tools which minimizes the experimentation and provides maximum information. They provides minimum experimentation and they requires minimum experimentation and provides maximum information out of those minimum experiment. And this helps in reducing the great deal of time, efforts, and resources. So the DOE approach basically relies on the Six Sigma principle. This particular circle indicates the define, measure, analyze, improve, and control. So DMAIC. So DMAIC approach is a Six Sigma approach principle. So the DOE approach or the DOE basic principle highlights altogether about the DMAIC, which is a Six Sigma principle. So initially you need to define your objectives, then you need to identify the variables which are measurable. So which is measurability of the variable is important. So there are some quantitative variable, there are some qualitative variables which can be associated with the product, but really we need to identify the quantitative variable which we can at least measure. Then after identifying the measurable variable, we need to link the variables, the input variables with the output variables, or even the factors with the response variable, and we need to analyze the impact of this impact between them. So that is how the actual scenario takes place with the help of the DOE approach. Then improvement can be takes place at any stage of development to ensure the continuous learning and improvement. So key to FBD are the experimental designs, and experimental designs are of various types. For example, this particular slide indicating so many examples of experimental design: factorial design, central composite, box banking design, simplex mixture design, optimal design. So these particular designs are basically used for response surface analysis, response surface optimization, while fractional factorial design, the Taguchi design, and the Plackett-Bowman design, these are used for the screening process. So whenever we are interested in identifying the variable which are influencing for a particular product, the exercise is called factor screening exercise, and we need to embark upon the screening designs, basically the low resolution experimental design like the fractional factorial design, Taguchi design, and the Plackett-Bowman design are called the screening design, while the high resolution design like response surface designs include the factorial design, central composite design, or box banking design. They are high resolution experimental design, and these high resolution experimental designs are used for response surface optimization. So these are the various cubic depiction of the experimental design which are used for systematic optimization of the drug products. So here I have given the example of full factorial design, fractional factorial design, central composite design, box banking design, optimal design, and Taguchi design. So in a 1 hour or 2 hour presentation, it is very difficult for me to explain about the nitty-gritty of this experimental design. Certainly, it requires a day-long or 2 days workshop kind of thing to explain the nitty-gritty of the experimental design. So that requires a lot of time again for understanding the nitty-gritty of the experimental design. But here I need to highlight more about the case studies. Here I need to highlight more about the case studies. So the step 4 of the experimental design is the modelization and validation of design and control space. So what exactly I mentioned in the step 4 of the ladder of 5-step methodology. So this is the step 4 where the modelization and validation of the design and control spaces are carried out. And step 5 is the identification of design space. So here is the concept of design space. I will take just a couple of seconds to explain more about the design space and how design space is actually important in understanding the concept of quality by design and how it provides regulatory flexibility. So initially the exercise of quality by design is carried out in the explorable space region. So entire explorable region involves the domain where we can work with specific knowledge, and within the explorable space the knowledge space is demarcated, and within the knowledge space the design space is identified through proper experimentation, through rational justification of the the ranges of the factors that we are using that will ultimately provide the ranges for the design space within the knowledge space, and within the design space the control space can be demarcated further as a in-house approach of controlling or tightening the design space further. So design space is a multidimensional interplay of various material attributes and process parameter. So design space is very important requirement as far as generic product approval is concerned and even new product approval is concerned. Movement within the design space is considered as no change, while the movement outside the design space is considered as change. That's why the identification of design space and selecting the optimized product within the design space provide a lot of regulatory flexibility, and the regulatory agencies like FDA, EMA, MHRA look for the design space identification in a right way so that the scale up approval changes after filing of the generic application or new product application can be minimized. This will reduce the time of approval. This will reduce the time for reaching the medicines to the market also. So design space depend upon the scale dependent parameters, scale independent parameters. So there are scale dependent parameters whenever the process changes from R&D scale to the production scale to the commercial scale or exhibit scale. The scale of the instrument, the equipment, the size of the equipment tend to change. That's how the geometric similarity and kinematic similarity of the equipments are required to avoid any deviation in the selection of the optimum formulation beyond the design space region. So this requires a lot of expertise for identifying the right design space, and the person who is basically at the helm of affair need to take care about of the geometric similarity and kinematic similarity of the equipments which are used during the commercial scale up cycle, during the commercial scale up cycle. So software aspect of using the design of experiment is not a panacea. There are so many software which are useful. So design expert, Umetrix, Minitab, JMP, and can be useful. So I basically prefer the design expert and Minitab for performing the experimental designs. And step 5 is the pilot plant scale up and production commercialization. This is more about the industrial aspect of controlling the things, where the control strategy basically identified at the end of design of experiment based optimization can be used for controlling the post commercial changes. Basically during the after the approval of product also, the annual review of product is carried out in the production level. So the annual product review dossier contains the key deviations that the manufacturer has experienced during the batch to batch manufacturing cycle. So that is comes under the annual product review, where the key control strategies that the manufacturer has adopted to stay within the design space is highlighted, and that's what the regulatory agencies need to see about the things. So QBD is omnipresent at all stages of product development. So QBD can be applicable to any product, not any product, and any type of delivery system or dosage forms even. So nanopharmaceutical products, I will take a couple of case studies from the nanopharmaceutical products. So nanopharmaceutical products we know at a glance mainly involves the lipidic system, the polymeric systems, the inorganic nanoparticle system, the viral nanoparticles, and the conjugated systems are also available. So nanopharmaceutical market is very huge, and the growth is expecting that the market share will rise nearly 47% in US market and other developed market, including the Asia Pacific region, Europe and Japan by 2025. So the investment with respect to the nanopharmaceutical product development is very high these days, and the global regulatory market, global healthcare market, basically focusing on investing the nanoparticle system, basically the nanopharmaceutical products in the nanocrystal, nanoparticles, liposomal formulation. These are a couple of formulations where more products are coming because of their robust nature, because of their scalability and feasibility with respect to industrial production is concerned. So I basically feels that the challenges and regulatory concerns are there in nanopharmaceutical product due to their complex manufacturing techniques, and high degree of product variability due to the complex manufacturing techniques and the complex nature of the nanopharmaceutical products. The variability is very high in nanopharmaceutical products too. And of course, the poor formulation stability is another concern, and development cost is again a burden for pharmaceutical industry to invest in nanopharmaceutical product. So altogether, uh, these parameters are responsible for challenges and regulatory issues associated with the approval and manufacturing of nanopharmaceutical product. So I will basically highlight upon the product variability, how nanopharmaceutical product variability can be reduced. So this is a systematic flow chart that the nanopharmaceutical scientist can implement, starting from the project initiation, the drug profile study with the help of reformulation aspects, and QTPP review, the CQ identification, the risk assessment has to be done very critically to identify the critical parameters um, which are more critically linked with the outcome of the entire exercise. So identification of CMAs and CPPs, and whether they are critical CMAs or non-critical CMAs, if it is a CPP, whether it is a critical CPP or non-critical CPP. So those things need to be considered, and followed by the design of experiment-based understanding of the product and process parameters. Influence on the end product quality should need to be there. And then the more or less this particular chart looks like the systematic approach of quality by design, and that's how the nanopharmaceutical product development should be there. This is an article lately I have published in Drug Discovery Today, Impact Factor 7.5 yearly. So this particular article mainly highlights the quality by design implementation exclusively for nanopharmaceutical products. So coming to the case studies, last but not least, I will highlight some couple of case studies that may be useful to you for actual implementation of the concept of QBD, and how the science and QBD implementation can really lead to a fruitful outcome with respect to the understanding and right implementation of QBD, that I will highlight in a couple of case studies. So the first case study is based upon the development of nano-emulsion system of a particular antihypertensive drug, that is olmesartan midazolam. So nano-emulsion system, all of us we know that the nanopharmaceutical system are the thermodynamically stable and isotropic system which contain lipids, emulsions, and co-solvent. So basically the drug which was selected here, the olmesartan, is an antihypertensive drug. It basically exhibits high oral bioavailability issue due to the poor solubility and lack of poor solubility and lack of permeability, and inherent capability for hepatic first pass metabolism. So nanoemulsion design here showed high probability for improving its oral bioavailability, and the initial nanoemulsion was prepared by identifying the quality target product profile. QTPP was identified. You can see the elements of QTPP like the dosage form type, the dosage form design. So the nanoemulsions are basically the immediate release systems, and oral formulation approach was used. Oral route of administration was used. So pharmacokinetic expected was short Tmax, and high AUC and Cmax was expected. So all these things are elements with respect to the target QTPP elements, and suitable justifications have to be provided for individual target elements of the QTPP. Followed that, the critical quality attributes are identified, and the critical quality attributes for nanoemulsion could be the emulsification time, the globule size, zeta potential, or even the drug release parameter or the permeability of the drug. So these things comes under the critical quality attributes of nanoemulsion formulation that I have prepared. So the Ishikawa fishbone diagram is a very useful tool, and Ishikawa fishbone diagram should be constructed, should be drawn in the very first stage of the quality risk management exercise. And the Ishikawa fishbone diagram, you can see that the 7M approach, that is men, material, machine, milieu, measurement, and methods, all these things were highlighted, and the critical parameters and sub-parameters were included in the fishbone diagram. So the main goal of preparing a fishbone diagram is to ensure that we are more concerned about all the variables or all the factors that a particular product may may get influenced. So we are not here to miss any single variable that can be vulnerable variable, that can be a critical variable in later situation, and we should not ignore those variables. So the ideal goal is to put all the variables in a one place in order to ensure that we are doing right from the beginning with some predefined objectives for maintaining the quality attributes of the particular drug product. So here is the case of nanoemulsion. So this particular risk estimation matrix indicates that the type of lipid, type of emulgent, the amount of lipid, amount of emulgent, altogether these factors are considered to be the highly risk factors, and they are mainly influencing the quality attributes of the nanoemulsions. So after identifying the risk estimation matrix, the solubility studies were carried out for identifying this the lipid, emulgent, and co-solvent with high solubility, and serotonin phase diagrams were prepared. And Taguchi design matrix was used as an approach for screening the formulation, for screening the nanoemulsion formulation, for selecting the quantity impact of the individual factors on the quality attributes. So various factors were considered, and the design matrix was identified with the help of Taguchi design. And this particular the design was executed, and the design suggested the role of these are the Pareto charts and half normal plot. So the design particularly suggested the role of the co-solvent, the surfactant, the lipids influencing the quality attributes of the drug product. That is the nanoemulsion. So after that, response surface optimization was carried out. Here is the design matrix for response surface optimization, and you can see that the 16 formulations were prepared for 3 different factors, that is the oleic acid, twin 40, and transcutol SP, and 6 response variables were taken. And this particular design matrix was further subjected to the response surface analysis. So here you will get the drug release profile under different conditions, that is using the dialysis bag with molecular weight cutoff of 1 kilodalton and 12 kilodalton. The 1 kilodalton initially experiment shows that the drug release profile was not very discriminating. So that's why the 12 kilodalton, the diffusion dialysis diffusion membrane was used for getting exact discrimination in the drug release profile from different nanoemulsion formulation which were prepared using the experimental designs. So these are some response surface plot, 3D response surface plots, and 2D response 2D contour graphs. So these graphs are basically the outcomes of the experimental design, and here exactly you can take the product and process understanding. So how the factors are influencing and which factor is influencing other factor. So the interaction among the factors can be studied using the response surface graphs or 2D graphs. So these 2D graphs are a little bit complex to understand, but yes, very useful, and you can really correlate the impact of one variable with another variable with these 3D and 2D plots, and that's how the product and process understanding is embarks upon. And FDA, basically the regulatory agency like FDA or MHRA, EMEA, they basically looks these things as the key important understanding that a formulation scientist has gathered with respect to the systematic product development has to be carried out. So this is a design space out of the mixture design which was used for the particular nanoemulsion formulation, and the flagged formulation indicates the optimum solution. You can see the design space, which is where the yellow color region well within the knowledge space region. And these are the validation charts, the linear correlation plots and residual plots indicating that the design and the model, the experimental model or mathematical model which was used for this particular experimental design has high degree of having the possibility of low degree of error or percent prediction error between the predicted values and the observed values. So I can bypass some of the slides because of the paucity of time. I have a couple of case studies more to show you. I will try to sum up within next 10 to 15 minutes. Yes, these are the particle size distribution profile and zeta potential distribution profile of the nanoemulsion formulations. And these are the 10 images of the formulation and rheological parameters and rheological behavior of the nanoemulsion. and these are the in vitro drug release profile for the cationic formulation. So nanoemulsion the plain nanoemulsion was converted into the optimized plain nanoemulsion was converted to the cationic nanoemulsion. So the particular charge inducer oleic acid was incorporated into the plain nanoemulsion to induce the cationic charge on the emulsion globule, because the cationic formulation is considered to be more potential in improving the bioavailability of the drug. And the specific purpose of using cationic charge was also to inhibit the role of bile salt absorption inhibitors, which basically reduces the absorption of lipidic formulation. And the nanoemulsion basically contains the triglyceride. So cationic charge inducer was also very helpful in reducing the impact of the bile salt absorption inhibitors in reducing the absorption of lipidic formulation. So some CACO2 cell line studies were carried out. The cell line studies indicated high uptake of the drug, and the drug which is encapsulated in the nanoparticles, nanoemulsions into the cells, and certainly the cytotoxicity studies were also carried out, and the cytotoxicity studies basically indicated that the safety of the nanoemulsion, and some more toxicity studies were carried out on rat hepatocyte with respect to the cationic safety of the cationic formulation, which indicated that there is a mild generation of reactive oxygen species on rat liver hepatocytes. These studies were carried out in our own laboratory at Punjab University at Jamia Hamdard both. So these studies basically indicated that the cationic formulations were having mild toxicity on the rat hepatocytes, but yes, these formulations were very effective with respect to the pharmacokinetic profiles of improvement in the Cmax and AUC of the drugs are concerned. So histopathology studies were carried out to check the safety of the formulation again on rat intestine, which indicated that there is no significant change in the undergoing underlying tissues of the intestinal region. So some perfusion studies were carried out to derive the permeability and effective change, the critical change in the permeability and the absorption behavior of the drug from the nanoemulsion formulation as well as the cationic nanoemulsion formulation. So all these parameters basically indicating the improvement in the absorption and permeability capacity of the drug. So some efflux studies were carried out. So basically the efflux is a basic PGP efflux is a major issue with respect to the nano formulation. So PGP efflux studies were carried out using some PGP efflux inhibitors. So inhibitors were used, and the role of inhibitor as well as the role of nanoformulation in reducing the PGP efflux was studied, which indicated that nanoemulsion were quite effective in reducing the PGP efflux of the drug as compared to that of the drug which was given along with the PGP efflux inhibitor. So plain drug given with inhibitor and the drug-loaded nanoformulations were compared with respect to their PGP efflux inhibition capacity, which indicated that the nanoemulsion loaded with drugs without administration of inhibitor also showed better results basically. So some other studies were carried out with respect to the calculation of the PGP efflux ratio with transwell assembly. So this work was later published in 2015 in IJP, and this work was basically appreciated a lot. So I will take one more case study before I conclude. I have a couple of case studies, but still I will take some case studies which are related to the other formulation. So I will take one more case study last before I conclude. So this case study is basically about the in situ gelling nanoparticle system of the onidazole. So onidazole is basically a antimicrobial agent. So it is basically recommended in periodontitis also. So in periodontal infection, the market formulation contains the drug solution. So drug formulations are basically considered as the solutions are considered as very less effective as far as the periodontal treatment is concerned, periodontitis treatment is concerned, because whenever the drug solutions are applied, directly into the periodontal pocket in the oral cavity, the retention of the drug in the periodontal cavity is very low. So for that purpose, in situ gelling system and incorporating the drug in nanoparticle can lead to very good results. That's what the hypothesis that we assumed, and we certainly prepared the PLGA nanoparticles of the drug. And sorry, not PLGA. These are the nanoparticles which are derived from the xanthan gum and chitosan. So sodium alginate and chitosan, and the formulation was optimized using central composite design. So these are the characterization parameters of the nano formulation, so which indicated that the controlled release profile of the drug was achieved with maximum entrapment till 70 to 90% from all the formulations that were prepared as per the central composite design. So these are the particle size distribution profile of the various formulations. You can see that there is a significant change in the particle size of the nano formulation or nanoparticles, with change in the polymer concentration, with change in the stabilizer concentration. And these are the 3D response surface plot, which indicates that with change in the polymer concentration, with change in the stabilizer concentration, the particle size and the release profile were continuously changing, and the factors were interacting. And this is the overlay plot, basically the overlay plot, which is indicating the optimum formulation. The yellow color region is the optimum region, and the flagged point indicates the optimum formulation with maximum entrapment efficiency, minimum particle size, and acceptable zeta potential and drug release profiles. So later, the formulation was compared for in vitro performance testing by salt to gel transformation studies. So as we know that the salt gel systems are quickly undergoes changes with respect to the formulation behavior is concerned. So in situ gelling was observed by the moment when the formulation was exposed to the hot distilled water at 25 degrees centigrade and 37 degrees centigrade. Both the pictures indicating the gel form of the solution. At 25 and 37 degrees centigrade, the formulation shows gelling, so which indicated that yes, the application of in situ gelling nanoparticle system to our to the periodontal cavity can lead to maximum retention of the drug in the gel form for prolonged period of time. So further, the in vivo pharmacokinetic studies in rat were performed in rats. So in vivo pharmacokinetic studies in rat induced with ligature. So the disease with the rat were tied with the ligature in for their dentine, and the rats tied with the ligature showed experimental periodontitis, and which were basically subjected to administration of the nano formulation as well as the plain gel. So comparison was taken place with plain gel nano formulation incorporated in gel and plain drug solution, and compared with the control disease containing animals. So these are the histopathological studies where it is indicated that showing that the nanoparticle drug loaded nanoparticle gel group showed maximum efficacy with respect to the disease, the periodontal infection reduction was concerned, and basically the plain solution showed less possibility, less efficacy for treatment of the disease as far as the periodontal infection minimization was concerned. So the marketed formulation was also used here in this study, where the nanoparticle formulation application for only 1 week was found to be superior over the marketed in simple in situ gelling formulation of the drug ornidazole after 3 weeks of application. So 1 week of nanoparticle, the novel nanoparticle formulation was considered to be much superior than that of using the 3 weeks of administration of the marketed formulation. So this was a remarkable achievement that the study provided, and later the clinical trials of the studies in actual human patients were conducted. So I cannot show you the human data because of the issue with respect to the patent filing is going on. But yes, but the data is promising with respect to the acceptability of the quality of the formulation and efficacy of the formulation in human application. And there is one publication which came out of this work. So these are the gamma scintigraphic images when the nanoparticles were compared with the plain solution and plain in situ gelling system. These are the gamma scintigraphy images of the rats treated with different treatment. So the third figure indicates basically the in situ gelling nanoparticle system in the periodontal cavity. So maximum nearly 6 hours observation indicated that the nanoparticles were still retaining in the periodontal pocket in the oral cavity, while in case of plain solution and plain in situ gelling system, the nanoparticles tagged with the gamma the radioligand were reached to the kidney region. So basically the elimination was already in the place, while in case of nanoparticle loaded in situ gelling system, the maximal retention was observed till 6 to 8 hours. That's what the study, initial part of the study has been published in AAPS, and there are some more case studies are there, but I will stop my presentation over here with the paucity of time. So further conclusion is it's all about conclusion. So here are some of the books. So I have there are some of the books which may be very useful to you on pharmaceutical quality by design, even the analytical quality by design and experimental design. So these two books were published very recently by Springer in 2021 January. So design of experiment for pharmaceutical product development, volume 1 and volume 2 are now available on Springer site. So these two books can be very useful for understanding with respect to the practical basics and fundamental principles of DOE are concerned, and the volume 2 indicates the application and practical case studies. So that's all about from my end. So if you have any question, then I will be happy to answer. I will be happy to discuss further after this presentation. Also, you can write me email so that we can discuss more about the concept of the quality by design and practical implementation of DOE are concerned. So
Sir, we have received some questions on chat box, sir.
yeah. When and who designed the experimental design protocol? When and who designs the experimental design protocol? So it's the formulation scientist. So the formulation scientist or the individual formulator is responsible for designing the experimental design protocol. So initially you need to carry out the pre-formulation exercise based upon your understanding with respect to the pre-formulation studies. You can identify the critical variables, critical factors, and you can establish the critical quality attributes so that you can generate your own experimental design model using the software which are available to you. Like the design expert is very user friendly, even Minitab is user friendly, and both software can helpful in providing you the right experimental design solution. Can we integrate kappa along with principles of QBD? Yes, kappa is always there. So implementation of QBD can lead to a control strategy. So control strategy is the place where the corrective and preventive action which are observed at later stages of product development can be incorporated. So I feel that yes, the right use of kappa can be there at the end of the product development exercise or even the analytical development exercise, where we certainly reach to some conclusion with respect to the challenges that we have observed, with respect to the understanding that we have observed out of DOE, out of experimental design. So the use of kappa can be there in designing the right control strategy. So use of kappa can be useful for continuous improvement during the learning cycle of entire product development.
Okay, thank you, sir, for your nice presentation.
Welcome, sir. Thank you for the opportunity.
For the recent trends, what is going on in the research for the faculty members and research scholars, both will be benefited for this. Thank you, sir, once again.
Welcome. Thank you so much.
Thank you so much, Dr. Beg, for taking the time to speak to our participants about concept of generic drugs and QBD, which are especially need of the hour. We truly appreciate young minds like you who are willing to share their talents to enrich the lives of our young researchers. May I now request Principal Professor B.V. Ravikumar Sir, coordinator of this event, to present the e-certificate of appreciation to Dr. Sarwar Beg, Sir, as a token of love and affection from entire RIPS family.
Rajaram sir.
Sir,
Beg sir. Thank you, Dr. Beg,
Yes, sir. Thank you, sir, so much, sir.
for accepting the invitation and sparing your valuable time for all of us.
Thank you so much, sir. It is a nice
and
opportunity
whenever-
to talk to all of you and present before you.
Yeah, whenever RIPS thinks about training program or faculty development program, your name comes first. And thank you for
My
all.
privilege, my privilege. It's my privilege.
Congratulations to you on your recent awards and medals.
Thank you so
And
much, sir.
welcome back to teaching profession again.
Yes, sir.
The one
Thank
you
sir.
are enjoying most. And on behalf of Roland Institute of Pharmaceutical Sciences, please accept this certificate of appreciation.
Thank you so much, sir, for all this arrangement and opportunity to speak on this prestigious platform of FDP sponsored by AICTE. So I wish you all under your leadership that such program uh, should be there at every year, so that participants in the faculty members from Roland and the nearby institutions in the vicinity of Odisha and the Andhra Pradesh, Chhattisgarh, they can be benefited, because the Roland has already been nominated by UGC as the nodal center for
Very
research
hard.
for PhD program. I basically I feel congratulating you. I am congratulating you for this great achievement under your leadership, and under the leadership of the management. The Roland is moving towards the excellence of research and education.
Thank you, Dr. Beg.
Thank you, sir.
from every ending, there comes a new beginning. With this, the session 2 of Day 1 Series 2 online FDP program comes to an end. We will resume the program tomorrow on the same platform. Until then, stay safe and stay connected with us. Thank you all the participants for your active participations.